Mhm. So good morning, all of you. I welcome all the participants, distinguished fellows, on the third day of AFM 2020 on behalf of Organizing Committee of AFM 2020. Today, I welcome Dr. Amia Priyam, Associate Professor, Central University of South Bihar, Gaya, to chair the session, first session of the day. So Dr. Amia Priyam, please take over.
Hari Srikanth, welcome to this. Good morning.
Good morning.
Professor Srikanth.
Good morning. Hi. Can you hear me? Is the audio clear? Okay, thank you.
Welcome to in the webinar, and many thanks accepting our acceptance. And
my pleasure.
Nice to see you in the morning, and you have taken pain in your place. This is exactly 12 o'clock in the night.
Yes.
So thank you very much. I request Amia Priyam to start the chair. Thank you.
Thank you, sir. I welcome you all to AFM 2020. All the participants, a very good morning to all of you. Here we have a distinguished professor from University of South Florida, Professor Srikanth, and he has hello, yeah. So he has an illustrious career, and he did his bachelor's and master's from Madurai, and then went on Madurai College, and thereafter PhD in 1994 from Indian Institute of Science, Bangalore, and right now he thereafter he had he has been at various positions at different institutions. Important among them is the Research University of New Orleans, and then USF Tampa. He's already there. He was a postdoctoral fellow at University of Nebraska in 94-95, postdoctoral researcher in Northwestern University, Boston, as well as national research. He is also a member. Sorry, he has. If I look at his Google Scholar profile, it's amazing. I mean, he has more than 400 papers, and H index is 51. He has also produced numerous PhD scholars, and he has his career is also studied with several achievements. In many things, he has several awards such as Marie Curie Fellows, and he's also Fellow of American Physical Society and Fellow of Institute of Nanotechnology. He is also an associate editor of Journal of Applied Physics. So there are many more things which can be said, but I know that audience is eager to listen to him. His research is basically on magnetic nanoparticles, and today's talk Today's talk, he will be talking about some biomedical applications of nanoparticles. So let's start. Uh, Professor Shrikant, we welcome you, and we are eager to listen to you.
Thank you, Dr. Priyam. Hello, Bhubaneswar. Good morning. I can't speak Odia, but I can say one word that is pretty much universal in most Indian languages, Suprabhat. Okay, so with that, let me first thank Dr. Parashar for inviting me to this conference. I looked at the agenda. I tried to listen to a couple of them. As you can tell, it's midnight here, so the timing is not quite right for me to stay late. And this is the first week of classes for us here. So it's been a pretty hectic week here in Tampa. So let me first say that I'm pleased to be here. I was in Bhubaneswar back in December 2018. But first, let me check. Am I clear? Is my voice clear?
Yes, yes.
My audio clear? Okay. All right. Thank you. So what I'll do is that, first of all, I'll share my screen, and then we can go ahead with that. It's a pleasure to be here again. And so let me go ahead and share my screen. All right, so you should be able to see my colors of my university, which is the green slide.
Yeah, we can see.
And you should be able to see its full screen now. Let me just quickly. All right, so again, as I said, I don't speak Odisha, so if you find any mistake on some of the script there, that's blame Google Translate. Okay, that's what I used. As you can see here, I want to talk about functional magnetic nanoparticles for biomedical applications, and specifically, and that's what it's supposed to say in your language, it's related to cancer treatment or cancer therapy. So specifically, that's what I'm going to focus on today. I would like to, again, thank the organizers, Dr. Parashar and others who are organizing this wonderful conference for inviting me here. And basically, let me get started by saying that this is a story. I'm going to tell you a story, especially of anisotropy. Anisotropy is very important in many materials, including ferroelectrics. I know that there's a strong background in ferroelectrics and multiferroics in this conference. In fact, I can recognize some good friends and people that I recognize. I see that Professor Ram Katiyar has given a talk in your conference. I also see a good friend of mine, Dr. Shavira Moya from Cambridge, who's talking today. But very important, I see this young faculty member at IIT Kharagpur, Dr. Prasanna Kumar Sahu. He is one of us, by the way. He did a postdoc here at University of South Florida in my department. It was not my postdoc, but one of my colleagues' postdoc. So we do have a good connection now with this group. Okay, so first, I can tell you that I've been to Bhubaneswar recently. These are pictures that I took in your famous Konark temple, the Sun Temple in Konark. This was in December 2018. And I would like to, show that, saying that, okay, I've been once in my life, I've been to Bhubaneswar and to Odisha in general. So I look forward to visiting again at some point. I'm looking forward to the good old days when we can travel freely, maybe 2021. We'll look forward to that. Okay, so let me tell you where I come from. I am from the city of Tampa. It's in Florida. And this is a map of Florida. You can see that Tampa, I changed it to a laser pointer right there. It's on the Gulf Coast, on the west coast of Florida. This is the Gulf of Mexico. And if you were following the news, you saw that there's a major hurricane that just hit this morning, yesterday. And that hurricane went right there, luckily. Okay, so we are quite far away. We were from the path of the hurricane. But usually we get a lot of hurricanes in this area. So that's one of the things that's scary in summer. But the beautiful beaches here, okay, these are some of the best beaches in the country. And I know that in Odisha, there are beautiful beaches too. I've been to one or two, Puri and many other places there. So we have some really nice beaches in Tampa. And in fact, it turns out that the Gulf Coast beaches are some of the best in the United States. And this is not just me saying about my city and my area. There are magazines that rank the best beaches in the country. And always Sarasota, which is south of Tampa, ranks first. Okay, so let me also tell you a little bit about my university. I would like to welcome you all to the city of Tampa and USF, University of South Florida. We are a pretty large university, and I'm trying to see if I can get this box here, minimize, so that's better. Okay, so I would like to point out that there are many attractions in the city of Tampa. Let me say a few words about my university. We are the 12th largest in the country in terms of public universities. There are about 47,000 students. And I understand that this, this conference is being organized by the physics department at KIIT, and you offer an applied physics flavor. So we do that too. It turns out that we offer the only applied physics PhD program in the state of Florida. When you think of Florida, you may have heard of other universities like University of Florida, which is in Gainesville, Florida State University in Tallahassee, University of Central Florida in Orlando. All of these are major universities with over 45,000 students in these campuses. However, as far as applied physics is concerned, we are the only physics department that offer this program. So again, I would like you to pass the word. We would love to have some students from Odisha join our department. If you'd like to know more about us, more about the different areas of research, you can go to the department website here and basically get to know more about the program. If you are interested in doing getting a PhD in applied physics, and that too near some of the world's best beaches, then we are the only choice here in this part of Florida. So please pass the word around. Okay, so I would also like to acknowledge my entire group. By the way, as you notice, my lab is called the Functional Materials Lab. So it looks like I am part of a family here because your conference is Advances in Functional Materials. So I fit right in. Okay, so again, we are a pretty large group, and these are some students right now. The ones in green have recently graduated and moved on. These are the current students, PhD students, all of them. We have some postdocs that have contributed to some of the work. These are some of the postdocs over the years. Many of them have contributed to the work that I'm going to talk about today. My colleague, Fawn, is a great collaborator, and he's a research professor in the department, and we jointly manage some of the group activities. These are some of the areas that we work in, and most of them are centered around different aspects of magnetic materials. So today's talk is going to focus mainly on this topic right here, which is application of magnetic nanoparticles to nanomedicine, or specifically to cancer therapy, as I mentioned. We are a magnetic materials and measurements lab, and it turns out that we do have a lot of facilities that can be used for various experiments. These are the kind of activities that we can do in my lab. Some of them are very specific. Some of them are general experiments. So today, I'm going to briefly mention this transverse susceptibility, which is a rather unique way of measuring switching and anisotropy in magnetic materials of any kind, whether they are nanoparticles or thin films or bulk materials. And then the other aspect is this one right here. SAR is an important figure of merit in cancer therapy. It's called the specific absorption rate, SAR. And that is used to measure the heating efficiency of nanoparticles for heating cancer cells. In fact, I'll present to you today that we can actually come up with a therapy where we can use magnetic nanoparticles and heat them up locally to heat up the cancer cells and cause their destruction. So that's one of the techniques that I will present today. So I'll tell you how we do the measurements, how we evaluate the heating efficiency of these nanoparticles. Okay, so this is the outline of my talk. I'll start off with a very basic idea about magnetic anisotropy. I'm a physicist, so there'll be at least one equation with one slide with equations. So please bear with me. I mean, it must be the midnight effect. I'm mixing up my words. Okay, so that's going to be, to start with, a fundamental introduction to anisotropy, why it is important, and in the context of nanomaterials, what does anisotropy mean and how we can use it. And then the main part of this talk is going to be utilizing anisotropic nanoparticles for biomedical applications. This one right here, I don't know if any of you have seen or heard of there used to be a popular TV show in the United States. It's called Dancing with the Stars. So I made a little word play on that. So today I'm going to talk about dancing with the SARs. The SAR that I mentioned is the figure of merit, which is specific absorption rate. I'll introduce all this so you don't need to worry about not following what it is. I'll try to make this talk sort of broad so that it appeals to a broad audience. Okay, so let's talk about anisotropy. And as I said, I'm a physicist, so here's a set of equations, and I'll show you what they are. So when you think of anisotropy, when you apply a magnetic field, and this is, let's say, a ferromagnetic material, and I know that most of you are interested in ferroelectrics here in this conference, probably. Consider ferromagnetism is very similar to ferroelectricity. Just replace electricity by magnetism, okay? And more or less, you get similar kind of behavior. You get the hysteresis. Instead of the polarization versus electric field, what we look at is the magnetization versus magnetic field, okay? So essentially, some of the physics is similar is what I'm saying. So when you apply, to understand magnetic anisotropy, let's apply a field, a magnetic field, on a magnetic material, and that's the internal magnetization. Of course, to understand magnetism, you have to go all the way to quantum mechanics because magnetism is rooted in quantum mechanics. We can't do that. We don't have that time. So let's start with the effective magnetization of this material, which is that one right there. So when you apply a magnetic field, this magnetization vector is now feeling two different energies, okay? So one is the internal magnetic energy, which is determined by the magnetic anisotropy. That's one of the primary contributions to the internal magnetic energy. And the other one is due to the external field. So we can represent this by the total energy here as a combination of two terms. One is the Zeeman energy, which is due to the external field. That's this first term right here. And the other one is, in this case, is a very simple uniaxial anisotropy. I'm just saying that there's no preferred direction. And that is the anisotropy energy. So you can see that the total energy and essentially the equilibrium position of this magnetization vector is determined by minimizing this. And you know how to minimize this, right? Students can do this all the time. And that determines essentially the direction of the magnetization. And as you can see, it's a competition between two important energies. When you apply a magnetic field, you have the Zeeman term, and then you have the anisotropy. And that's exactly what results in the shape of the magnetization curves. You see that you have an easy axis if you can magnetize it very, very easily at very small fields, then you have a shape that looks like that. And if it's hard to magnetize it in a different direction, of course, because of the fact that you have the anisotropy energy being dominant, then you see that the shape is something like that. So this determines the easy direction, the hard direction, soft magnets, hard magnets. There are many applications depending on the shape of the MH curves of the so-called hysteresis loops, okay? Well, this is just the first quadrant, right? If you do the full loop, then you can see the hysteresis loop, which looks very similar, which would be looking very similar to the ferroelectric hysteresis that many of you are familiar with and probably heard about, saw a lot of them in this conference. Okay, so now that's the uniaxial term is simply saying that you don't have a preferred direction. You can actually include the preferred directions. You can take other contributions such as cubic anisotropy. However, when it comes to nanoparticles, there are many terms that can contribute to the anisotropy, okay? Let's start with the this one right here, and surface, okay? Well, it turns out that nanoparticles are all about surfaces. It doesn't matter if it's magnetic or metallic or quantum dots, whatever they are, the fundamental properties, the physical properties are governed by the surface atoms. So anyone working in nanomaterials is essentially a surface scientist. They're doing surface science by engineering the surface of the nanoparticles, okay? So one can actually do that, and I'll show you examples how we do it. We can actually play with the surface of nanoparticles and thus change the surface anisotropy and then change the shape of the hysteresis loop. The shape of the loop is key to our application, which is the heating efficiency for cancer therapy, okay? I'll get to that. So we, as physicists, we have to try to come, or material scientists, we have to try to come up with ways to change the shape or tune the shape of the hysteresis to improve the heating efficiency of these nanoparticles. So we can do that by various methods. Surface shape is easy to understand. You can make nano rods, and there's a preferred orientation. For example, the longitudinal direction, where you can easily magnetize it. So there's a distinct shape anisotropy there, which you can control. I'll show you how we can make these nano rods, and they're very important, elements for this application. Strain is important because you can have, for example, thin films. You can have a substrate which is different from the magnetic film on top, and the strain at the interface can result in contributions to the anisotropy. This is the magnetoelastic contribution, and that can be used to tune the shape of the hysteresis loop. Configuration, you can make nanoparticle clusters, and these clusters are attached to each other. You can make them in different sizes and shapes and configurations. So that can be used for playing with the anisotropy. And last but not the least is this exchange anisotropy or unidirectional anisotropy. Now, when you hear of exchange bias, this is something very important for the spintronics community. Those that work in magnetic materials, MRAMs, spintronic devices, for example, they're really, one of the critical properties is the interface between a ferromagnet and an antiferromagnet, okay? If you have an interface between an antiferromagnet and a ferromagnet, it turns out that because of the differential anisotropy at the interface between these two magnetic phases, the interfacial spins are pinned. And so you can have very interesting consequences in the sense that the coercivity of the hysteresis loop can increase, and also the loop itself can shift. This is known as the exchange bias. You can actually have the bias field, which is determined by the shift in the hysteresis loop. So that's an important contribution of interfacial anisotropy in playing around with the hysteresis loops. And today, I'm going to tell you that exchange bias, which is very important for spin valves and MRAM for the spintronic devices community, is equally important for the nanomedicine community, because we can actually use the exchange bias between the core and shell in core shell nanoparticles and use that for changing the characteristics of the magnetization. All right, so these are the things that I will introduce today. Okay, so I talk about anisotropic nanoparticles as if they're exotic. Actually, they're not. They're everywhere. They're ubiquitous. Okay. In fact, it turns out that making perfectly isotropic nanoparticles is more challenging than making anisotropic nanoparticles. So to give you an idea why anisotropic nanoparticles are universal, I'm going to show you 3 TEM images. These are transmission electron microscopy images. It's the same material. These are all iron oxides, magnetite, Fe3O4. There are other phases, gamma, Fe2O3, which is called magnemite, which is also commonly used. These are all spinel ferrites, okay? So all of them are the same material, ferrites, but where they come from in these 3 images is what makes them interesting. For example, let's start with the one here on the left. Now it turns out that these are actually magnetotactic bacteria. These are organisms, they're magnetotactic bacteria that actually produce magnetite nanocrystals. You can see the scale bar here. That's 250 nanometers. So these are beautiful, 20 nanometer particles, and they're chained up here. They're chained up by to form these short chains using dipolar coupling. And essentially what these bacteria are doing is that they're making tiny nano compasses, because you know that a compass is a needle, right? A needle that is basically pointing towards the north, right? As people, navigators would use that. So these bacteria are doing exactly what the Chinese did when they discovered, I know China is a bad word now in India, but anyway, so this goes back ancient times when the Chinese are credited to have first discovered the first magnetic material known to man, and that is called lodestone. Lodestone is nothing but Fe3O4, magnetite. Okay, so these are actually magnetite nanoparticles, and the bacteria are doing exactly what the Chinese did. They're actually making compasses and use it for navigation towards food. That's what they're doing. Okay, but see the biologically built-in anisotropy. They're making these dipolar coupled chains, which is highly anisotropic. I just wanted to illustrate that naturally anisotropic particles can be assembled or can occur. The second example, this one right here, is out of this world. It's not just an expression, it is literally out of this world. So what you're seeing there is the same magnetite, Fe3O4 nanocrystals or nanoparticles, but these are actually found in a chunk of meteorite from Mars that crashed into the Arizona desert many years back, okay? So obviously, one thing that comes to mind is you got magnetite nanoparticles in life forms, magnetite nanoparticles in rocks from Mars. So is there life on Mars? No, we're not going there. Okay, that's not the intent. I just wanted to show you that how anisotropic these particles are. Look at the faceted surfaces. Very important and interesting. They're all anisotropic and they form naturally in this process in the meteorite. Okay. And the third one is man-made. These are actually, if you look at the similarity, they're the same materials. They're ferrites. Similar to that, they're also faceted with interesting facets of the surface. And these nanoparticles are man-made. They're actually produced in a plasma chamber at a lab of a good friend of mine, at Michael McHenry, Professor McHenry at Carnegie Mellon University in Pittsburgh. So they use a microwave plasma technique to make large quantities of these anisotropic nanoparticles. So I just wanted to tell you that anisotropic nanoparticles are interesting and they're there everywhere. So let us use them for biomedical applications, for example. So that's what I'm leading to here. Okay, so I'm going to skip through this because, again, I'll be digressing from the main topic if I spend more time here. But what we did, and just wanted to plug a little bit about our contribution in this field of magnetic anisotropy, we were one of the first groups that developed a very sensitive resonant technique for measuring the so-called transverse susceptibility. We do this measurement in a slightly different way. This is a resonance circuit. And again, I don't have time to describe this, but essentially because it's high frequency, it's done at megahertz. So essentially it's like an FMR measurement, ferromagnetic resonance measurement. And from that, we can extract the change in the transverse susceptibility. And there's a reason why do it transverse. By transverse, you look at this one right here, the DC field is perpendicular to a small RF field on the sample, making it a perpendicular geometry. The transverse geometry is not necessarily very unique because that's exactly what is used in FMR measurements or even in NMR, nuclear magnetic resonance involves a DC field and a perpendicular high frequency RF field, okay? So it's very similar to those techniques, and we can extract the transverse susceptibility or the change in transverse susceptibility. And again, there's a very famous paper by Amika Maharoni, who's a well-known theorist in the field of magnetism. And he had predicted that for single domain nanoparticles, you can actually see these sharp peaks. He calculated the transverse component of the susceptibility, and he showed that these peaks here can occur exactly at the anisotropy field. So if we can do these experiments, exactly what we did. So what we did was we actually confirmed Aharoni's theoretical model, 40 years, 50 years after he proposed it. Okay, so that's why our experiment sort of became quite popular in measuring anisotropy, because yes, we can detect these peaks. These are the Aharoni peaks that he theoretically predicted, and we can measure this with our data that I showed you using the experiment. We can follow that as a function of field, and we can do it at different temperatures. So we can get an entire phase diagram in the HT plane for nanoparticles. So here's, for example, for 20 nanometer nanoparticles of magimite Fe2O3, all the way from high temperature to low temperature. So you can see that that's the blocking temperature right here. So you have a super paramagnetic response at high temperature, and then this blocking here. And you can also see how these peaks, the anisotropy fields, evolve. Since anisotropy field is one of the most fundamental and important property of magnets, right? So we have a very unique and quick way of rapidly screening different kinds of nanoparticles and looking at the switching and anisotropy fields. So that's the importance of this technique. Okay, so far I've told you that nanoparticles can be used for various applications. I told you that anisotropy is important. I even told you how to measure it accurately. Now let us see how we can use these anisotropic nanoparticles for biomedical applications. So the remainder of my talk is going to focus on the biomedical applications of these nanoparticles. Okay, so let's see, for example, where the magnetic nanoparticles are being used these days for many biomedical applications. If you start from the left here, targeted drug delivery, okay? So it turns out that if you can take these nanoparticles and you obviously make them biocompatible by doing some surface functionalization, and then you can inject it into the human body or into an animal, and using an external field, because it is magnetic, non-invasively, you can actually move it to an area where you want the drug to be delivered. For example, you can take the nanoparticles and you can coat them with drug molecules and then locally guide them with external magnetic field to deliver the drug. So in this case, magnetic nanoparticles are like ships. They're like navigational aids for taking or transferring the drug inside the human body. So this is an area, there's a lot of work. We don't work in this field, but I just wanted to mention that targeted drug delivery is an area where a lot of people are working. And why is this important? Well, think about it. if you have any affliction, for example, well, these days it's all COVID-19, but there are many other diseases, as you know, cancer being one of them that we need to tackle all the time. And so basically, when you take a medicine for something, mostly you take a pill, you swallow it, or you take a liquid form, right? And that is going through your entire body. In other words, it's coursing through your the central system, right? And essentially what it means is that your whole body is getting exposed to this drug, and there are side effects. So the idea of going to targeted drug delivery is that you expose only the part that you need to treat, not the entire body. And so this is more effective and there are less side effects. So that's the idea behind local or targeted drug delivery. A second area where there's a lot of effort being done and magnetic nanoparticles have made a huge impact is that you can use these magnetic particles for improving the contrast in MRI images. MRI is magnetic resonance imaging. It's very commonly used in hospitals where basically it's based on NMR. When you do NMR, water's most of the tissue, biological tissue contains water. So you can actually excite the water protons, let them process by applying a DC and high frequency oscillation. Then you remove the field, they relax. And from the relaxation time, you get images like these. This is this way from the T2 relaxation time, you can get these images, and that's your standard MRI image. Now, it turns out that if you inject magnetic nanoparticles into the brain or whatever tissue you want to do the MRI on, these magnetic nanoparticles have their own fringing fields which interact with the particles, with the tissue, with the external field. So because of this, it changes the relaxation time, T2 relaxation time. And because of this change, you can actually develop a higher contrast. So it has been demonstrated that nanoparticles can be used for MRI enhancement. There's a new breakthrough in this field. This is called magnetic particle imaging as opposed to magnetic resonance imaging. In this case, the particle itself is imaged, whereas here it's the water protons, right? So you're doing it indirectly from the NMR. In this case, the particle itself is imaged, and you can see the beautiful contrast. On the left here is a mouse with MRI, conventional MRI with nanoparticles. On the right here is the same image now done using MPI, or magnetic particle imaging. This came out of Berkeley a few years back. Now it's very popular. And you can see that the contrast is beautiful. It's amazing. So this is the future of magnetic imaging of tissue and brain and so on and so forth. But the main focus of my talk that I'm going to focus on right now is using these magnetic nanoparticles for hyperthermia or heating. In other words, instead of attaching drug molecules, can we use the particles themselves to kill cancer cells? And the answer is yes, we can do that by locally delivering heat, toxic amounts of heat. That's what I'm going to talk about now. Okay, so again, when you talk about nanoparticles, this is a nice slide that captures from this paper right here, that captures pretty much what is involved here. You need to first choose the type of nanomaterial. There are plenty out there. It could be biomaterials, metallic particles, carbon nanotubes, so on and so forth. And then, of course, you have to look at the size range that you need to focus on. Depending on the application, you choose the size. This is most important. Today, I'm going to focus on this, the morphology, or in other words, anisotropy, the shape, so on and so forth, can be used, and that can be also tuned. And then finally, remember that these nanoparticles have to be functionalized. You have to do a lot of surface chemistry. You have to make them biocompatible, for example. You have to attach antibodies, peptides, because you can't just inject these particles, because obviously there's a big war going on in your body, and the body will reject these particles. So you have to coat them, essentially sugar coat them, literally, because you can actually use dextran and sugars and glucose to coat these particles to make them biocompatible for the cells. Okay. All right. So let's talk about cancer. This is 2017. I don't know why the top keeps popping down, but anyway, so this is basically data from 2017 that shows you in millions how many people are generally affected by cancer. India is right there. It's blue. So there is a large number right there. And I got the cancer statistics. If you go to cancerindia.org.in, you can find the most recent statistics here. So if you look at this right here, There are estimated about 2.25 million people right now at any given time, 2.5 million people that are living with cancer, okay? And these are stats about 11.5 lakhs, and cancer-related deaths on average is close to 8 lakhs per year, okay? So this is basically, these are large numbers, and essentially we need therapies of various kinds to control cancer. Again, the focus is on COVID-19. Of course, it's important to try to find the vaccine and translate it to the public, which is on its way. But cancer has always been around as a major threat. And in fact, two of the biggest diseases that mankind, or basically that leads to deaths, two of the most intense or most high-risk diseases that leads to death are heart disease and cancer, number 1 and 2. Depending on how you look at it, one of them is first and the other one is second. Okay, so one question you ask is, well, cancer, what will I do? I'm a physicist, I'm an engineer. I mean, how will I work on cancer applications? The answer is we can. And to demonstrate this is Physics Today issue of July 2019, just last year. And there's an article here which tells you how physical and mathematical modeling has been very, very effective in understanding the process of metastasis. Metastasis is the process by which cancer cells grow and they leave their origin and then move throughout the body. One of the dangerous forms, because when cancer spreads, that's when it goes out of control, okay? So there are models now available, and guess who helped develop these models? Physicists and mathematicians. So yes, of course, as students, you can think of ways of using your skills, whether it's material science or physics or modeling or simulations, to tackle problems that are important to mankind, cancer being one of them, okay? All right, so how do we kill cancer with nanoparticles? The answer is simple. What we do is that we take magnetic nanoparticles and apply an oscillating magnetic field with large amplitude, okay? Because we need to completely go over the hysteresis loop again and again, okay? And at high frequency. And that causes heat, and that will destroy cancer cells. I'll explain to you the physics behind this. But first, I'm not a biochemist or a biophysicist, but this is what happens when these cancer cells are subjected to heat. Essentially protein denaturation occurs, and then the cell membrane restructures, and eventually it breaks or destroys, or the cell is deactivated, okay? The goal here is to destroy the cell membrane, and we have to think of ways of doing that. And one of the ways is to increase the temperature, and this is called apoptosis, heating it up and driving it towards its death in a benign way, okay? Sort of mercy killing of cancer cells, if you may think of that. And it happens at a very, not a reasonably high temperature. The hypothermia window where the cancer cells die is to expose them to a temperature just a few degrees Celsius above the body temperature. So we're talking about a window which is 40 to 45 degrees Celsius, okay? And when you increase the temperature of these cancer cells to this temperature range and hold it for some time, typically 45 minutes to an hour, that's the treatment time for this, cancer cells die. Okay, so that's an effective way of killing cancer cells. Now, this process, as I said, is called apoptosis. The other extreme is this term called necrosis. Necrosis is a brute force, aggressive way of killing cancer. So are there examples of necrosis? Of course, sure we do. That's what we use in hospitals. This is basically your radiation therapy, your chemotherapy, which are extremely advanced, very effective in controlling cancer. But again, why is it necrosis? Because look at the damage, look at the collateral damage. You get a lot of side effects. Those of you that may have either your friends or family members that are undergoing cancer treatment, you've seen the visible changes they go through, right? Their body goes through, they lose their hair. I mean, that's not the only thing, they lose their immune system, immune properties. So essentially, it's a brute force way. And so what we're trying to do is also introduce magnetic hypothermia as a more benign way, as a supplementary technique, which would assist along with chemotherapy in controlling cancer. So that's the idea behind this particular technique. Okay, so why is it not out in hospitals yet? The answer to that is what we are going through right now with the COVID, right? Why is the COVID vaccine taking such a long time? Well, there are labs developing it. Why is it not out to the public yet, not in the hospitals yet? The answer is you need to do clinical trials. You have to be very careful whenever there's a new technique or a new drug. It has to go through several stages of approval by the FDA. This is the Federal Food and Drug Administration. That's what we call it here in the United States. And there are equivalent, offices and agencies in India and other countries too. So without approval, you cannot just, transition it without clinical trials to hospitals. And that's the stage that's going through right now with the COVID-19 vaccine. And similarly for these treatments. So it's a slow process, okay? Because of the risks involved, we're talking about human patients here. However, it turns out that people in Europe, they're actually pretty ahead in the game. In fact, in Germany, there's a company here which actually does this technique of heating up nanoparticles and doing it. They inject the nanoparticles, then apply the AC field and heat it up. So it's almost like an MRI machine there. And they call this the nanotherm therapy. So let's briefly talk about the physics of how this works. The answer is hysteresis loops. Why do you think I spent a lot of time explaining anisotropy and the shape of the magnetization curves? Because that's what we need. We need to maximize the hysteresis loop. Here's a strange looking hysteresis loop. This is M versus H. I know you've seen a lot of these cigar-shaped loops in ferroelectric materials, probably in this conference. This one looks like that, but it is not like any magnetization curve, right? This is strange. And the reason this is strange is because this is the AC hysteresis. This is the high frequency hysteresis loop. It is not the hysteresis or the magnetization that you measure in a magnetometer like a SQUID or a VSM, for example. That's the DC measurement, right? So this is high frequency hysteresis. So in other words, when you apply a high frequency, the magnetization is trying to follow it and it cannot, it goes out of phase. And because of that, you open up a hysteresis loop. But what we need to do is that's exactly what we want. That's why we need to apply high frequency, because we can maximize the area of the hysteresis loop. And remember, basic physics tells you that the area inside a hysteresis loop is nothing but dissipation. And dissipation here is in the form of heat. Power loss is in the form of heat. And that's how we generate heat, okay? So like in any new technique, we need a figure of merit. So the figure of merit that is used here is called the SAR, specific absorption rate, which is area multiplied by the frequency, okay? So what we need to do is we need to get a large SAR. At the same time, remember these nanoparticles are foreign objects, so we don't want to inject too many of them in the human body. So we need to come up with ideal materials where we can maximize the SAR, but keeping the number of particles, small, okay? So we need to do a lot of material science for that. Okay, so here's another slide. Again, I won't spend too much time. The power loss can be calculated, and you can see that it's a product of the AC amplitude and the frequency. So that's what is important here. And there are two different time scales involved. These are relaxation times. One is the Brownian, the other one is the Néel relaxation time. So it's a combination that matters. And essentially what happens is that SLP is the same as SAR. Some people call it specific loss power. You can see that if you increase the magnetization of the material, of the nanoparticles, then the SAR will go up. Remember, our goal is to increase the SAR, maximize it as much as possible. There's a size dependence, which is very important. We cannot work with small particles and we cannot work with large particles. The best particles are somewhere intermediate. They're close to the single domain limit, but not quite super paramagnetic. There's also an important health reason why we cannot use very small particles. And I'm talking about particle sizes smaller than 10 nanometer. There's a health hazard, an important health hazard, because if you make very small particles in the human, inject it into the human body, there's a high possibility of these particles penetrating the so-called blood-brain barrier and ending up going to the brain. Now that's the last place you want the particles to end up, right? If you have a cancer somewhere in the kidney, that's where you want the particles to go, not to the brain, okay? And it's dangerous to have the particles going to the brain. So essentially, one has to be careful about the choice of the size of the particles, magnetic particles, for these measurements. Okay, I need to move on quickly because I'm spending a lot of time here. So how do we measure the heating? So this is what we do in our lab. Sorry, was there a question? Okay, I thought I heard somebody. So basically, we do it in two different ways. In our lab, we measure the heating of these nanoparticles by using the calorimetric method. Here's the AC magnetic power supply. And that's the coil right there. We put the particles in there. We supply a large amplitude AC, and then we measure. What we do is that we stick in a temperature sensor here, a thermometer, and then measure the change in temperature as a function of time, and we can get the SAR from this relation, okay? Our data looks like this. This is temperature in degrees Celsius as a function of time. And as you can see, the hypothermia window is 40 to 45 degrees Celsius. So for these ferrite nanoparticles, we can play around with the AC amplitude. Our frequency of the AC power supply is fixed. It's 325 kilohertz. I'll tell you why these numbers matter, but it's fixed. So we can only change the amplitude of the AC. So these are for different amplitudes. And you can see, you can select the best combination of frequency and field where you get the saturation right there at the hypothermia window, because essentially you don't want to overheat it, right? If you don't, there's no need to overheat the particles. And of course, if you don't supply enough amplitude at the right frequency, you will not heat up at all. The saturation is because of thermal equilibrium between the particles and the surrounding tissue, okay? So you can tweak these conditions so that you can achieve the hypothermia treatment window, and you can select these for different particles of different sizes by playing around with these conditions. We also send these particles to our collaborators in Spain, and what they do is actually they measure the AC hysteresis loop. They actually measure the entire high frequency hysteresis loop. So they built their own capacitor banks because it requires large power, and they measure the SAR by multiplying the area times frequency. So for the same particles that we make in our lab, we do 2 different experiments, and we compare the SAR and make sure that we get a quantitative match before we take it to the next stage of application, okay? So remember, one thing to keep in mind is that for students, this is a good exercise. If you want to publish in a high impact journal, you would like to develop a set of data that is also reproduced by other experiments, okay? So it makes the case more convincing. And not only that, it can be from a different lab. It makes it even more convincing. So that's what we're trying to do here. However, it's not only a different lab, it's in a different country, in Spain. So basically that's the idea, to seek international collaborations, and material science is all about interdisciplinary international collaborations. Can someone tell me if I can take about 10 minutes more or so? Because I think I started a little bit late. Is that okay? The session chair, can you tell me if 10 minutes more is okay?
Yes, sir. It's okay. You can proceed.
Okay, thank you very much.
Yeah, you can go ahead. Go ahead, sir.
Okay, thank you. All right, so why do I keep saying a few hundred oersted and a few hundred kilohertz? I said this is the limit, and that's determined by this product. Remember, this is the most important thing. The product of the AC amplitude and the frequency in these units, don't worry about the number. This is like the pain threshold. Remember, this treatment is for humans, okay? So we have to make sure that since we are subjecting the patient to extreme conditions of high frequency and high amplitude AC, we cannot just arbitrarily apply any field at any frequency. Because if we do that, then you have the risk of nervous stimulation, okay? Nerve stimulations, it can lead to stroke. So essentially, you have to be very careful about the choice. So that's why we have to work with low fields and low frequency to the extent possible. We cannot increase to Tesla fields to gigahertz frequencies at these amplitudes, okay? All right, so now the only thing left is, well, the first thing is you can increase the saturation magnetization, right? But it turns out that iron oxide is low compared to iron or iron cobalt. And guess what? Our hands are kind of tied here by the FDA because only iron oxide is considered biocompatible. This is the only approved biocompatible magnetic material that has been approved by the FDA for this treatment. So we have to work with this one material. We cannot just arbitrarily make any kind of magnetic material with large magnetization. So we have to work with one material. Iron, cobalt, we cannot do that. But one thing you can see is that, can we make a core shell particle where we can have the core as iron, which has a larger magnetization, but a thin shell of iron oxide. So we have a core shell system, and essentially, will that be, will improve the SAR? So that's what we tried first. We tried to make core shell particles, and here's a TEM of a core shell particle. Iron oxide is 92 EUV per gram. That's the saturation magnetization. Iron is much higher. So we had iron core and iron oxide. Because iron oxide is coating the entire core, this whole thing is biocompatible. So we're trying to fool the cells by hiding the toxic iron by covering it with biocompatible iron oxide. So we looked at this, and those of you that are interested in chemistry, what we do is very simple solution chemistry for synthesis of nanoparticles. But it turns out that when we make these core shell particles, there is something known as Kirkendall effect. Because iron is a metal, iron oxide is an oxide, of course, the differential diffusion of the oxygen ion and the metal ion is very different, okay? And because of this, there's an instability at the interface, and you have these little holes, voids opening up, and eventually over time, the hole gets bigger and bigger. Guess what? You end up having a hollow nanoparticle. Basically, the shell increases because the iron oxidizes and becomes iron oxide. And essentially, you start off with a core shell, and over time, you end up with a hollow shell. So if your goal is to make hollow nanoparticles, this is the way to go. But unfortunately, it's not good for us because as a function of time, the material itself is converting from core shell to hollow. So which means the magnetization is continuously changing, and it's not a good system for us, reliable system for SAR. We did a lot of studies. This beautiful set of TEM images my student took to look at the progression of going from core shell, where the core is now becoming smaller and eventually hollow. Okay. All right, so let's look at the SAR, and I'm going to show you the results in histograms just for comparison, because histograms, you can actually see that the histograms, you can compare at different conditions. So this strategy of using a core shell particle indeed worked because of the larger magnetization of the iron core, the SAR. Remember, SAR is the heating efficiency that we measure using calorimetry or the other method, right? And you can see that in every case for different sizes, compared to the all solid iron oxide, the core shell particles definitely heat up much better. SAR is much higher. However, our happiness was short-lived because as a function of time, and we don't have any control over this time because it's a process that we cannot control. Over time, of course, it degrades, and when it becomes hollow, it doesn't heat up anymore. So we thought it was a great idea, but we had to discard it because of this Kirkendall effect, which is due to the differential diffusion of the metal ion versus oxygen ion. But we thought that the interface anisotropy at the core shell particle is the way to go, okay? Except that we should avoid Kirkendall effect. Now, if you think about it, the best way to do that is to have all oxide core shell. Namely, the inside core is also oxide, outside is the shell is oxide, biocompatible iron oxide, of course. And then, of course, you don't have this Kirkendall effect. So we were thinking about it, and there was a group in Korea that actually made this before us, before we did. But they used cobalt ferrite and manganese ferrite. They actually showed that this is exchange coupled. So because cobalt ferrite is a ferromagnet, manganese ferrite is an antiferromagnet. So it's like a nano spin valve, okay? It's an exchange bias antiferromagnet system core shell. They actually showed that this exchange bias can be seen. However, I took one look at this, cobalt. Cobalt is poison. You can never use cobalt-based materials for medical treatment. So essentially, we need to look for iron oxide-based, core shell approaches. And this is what we did. And I would like to credit my postdoc here. Her name is Hafsas Magic, and she's actually an assistant professor now in Sharjah in UAE. She's originally from Pakistan, but she was extremely clever in making chemically synthesized nanoparticles. And we call this Hafsas Magic or Hermione's magic. You need some kind of, it's wizardry that you need to exhibit in the lab to make very high quality nanoparticles. So we actually made this FeO, Fe3O4 nanoparticles. FeO is the core. And remember, the shell has to be biocompatible. So we have no choice there because FDA will approve only biocompatible materials. So this is Fe3O4 shell. So we've got the core shell particles. So let's look at the other thing was Hafsa came to me and she said, but do you want me to play with the shapes also? And I said, sure. I mean, it's already very complicated with core shell, but she was able to play with the shapes inside. You can see the core shell. Can you see? You can see the core shell, core and shell, but she was also able to change the surface anisotropy, spherical, cubic, and these star-shaped particles for the same system, okay? So that we can compare the effect of the shape anisotropy or the surface anisotropy. This is one of the most important slides in this presentation. Here's the comparison: spheres, cubes, octopods, or these exotic shaped. Just look at this right here. there is no question under the same conditions, as you increase the anisotropy, as it becomes a lower symmetry system from spheres to octopods, clearly the SAR increases. This is one of our main contributions from my lab, where we actually told them that, listen, you don't need to look at other materials. Even iron oxide, play with the surface anisotropy, change the shape, and you can actually enhance the SAR beyond the normal spherical particles that people use. We also worked with the chains. Remember the magnetotactic bacteria? We actually took the bacteria and measured the SAR of these bacteria. That was quite promising too. We made nano rods. Again, I'm trying to rush through because I want to come to the conclusions. We made nano rods, and we played with the different sizes and aspect ratios. Nano rods gave us some of the best results. And this work was done by Raja Das, who's another postdoc. It's kind of interesting because Raja is from India, as you can tell, is originally from Assam, and Hafsa from Pakistan originally, right? They were in the same lab, and both were amazing postdocs. And there was a friendly competition between them who would make the best particles. So for me, it was like watching an India-Pakistan cricket match. I enjoyed it, had fun. Both of them are great, and they were able to publish their results in very good journals. So anyway, the nano rods gave us some of the best results in terms of the largest SAR. Again, the message is anisotropy is key. You can play with the anisotropy even with the known magnetic oxide and increase the SAR. So I'm going to skip a few slides, point out that everything that I told you today in this talk is only half the story, because I talked only about the material science part. We also need to worry about the biochemistry, because they have to be biocompatible, the surface has to be functionalized, and we have to look at the toxicity. So we did toxicity studies by actually feeding these nanoparticles to different kinds of cells, including cancer cells. So these are macrophages. Macrophages are very hungry. They like to eat whatever they get, okay? And these are nanoparticles that you add to the macrophages, and then basically after 24 hours, you can actually see that they're all inside the cells. And then we do the studies, whether the cells can survive alternating fields, current with the particles, without the particles, and that's how we do the study. So let me just run this video. This is just to show you that particles being taken up by cancer cells. This is work from a French group, and the young lady who did a PhD last year, she asked me to share this as part of this presentation. And you can see that these are gold permalloy nanoparticles, and these cells are fighting to eat them. The cancer cells love to eat these particles, so let's feed them and kill them. They want to die, okay? So here's a way of using magnetic particles and the hypothermia treatment to essentially kill cancer cells. Very effective, and very soon it'll be out in the hospitals. It's only a matter of time and the process of clinical trials, and we are close to that. I would say that in the next 5 years or so, this should be a supplemental technique available in most hospitals, cancer hospitals, in addition to chemotherapy and radiation therapy. So with that, there are other ways of destroying cancer cells. Remember, the goal is to destroy the cell membrane. You can also mechanically destroy it. There are ways to do that. And there are some papers on that. So with that, let me conclude by saying that size does matter, both in English and ODS, okay? And essentially, I'm not talking about nano size. Nanoparticles, nanomaterials have been around for decades now. We have to go think beyond the particle size. We have to think about controlling the particle shapes, surfaces, interfaces. So those are the keywords for the next generation. You students working in nanomaterials, start looking at shapes, the role of shapes, surfaces, and interfaces. This is what is going to determine, technology in many, with nanotechnology in many aspects, not just magnetic, but in other areas too. So I'm the editor now of this journal, Journal of Alloys and Compounds. So do send us some of your best papers. So some pictures from my lab, students, taking measurements, various systems that we have. And on behalf of my functional materials lab at USF, I'd like to thank you for your attention. On the left, you see the good old days when we used to hang out and party in the evenings. And of course, on the right, it's now COVID days. So we have to follow a lot of protocols in the lab, face masks, social distancing, so on and so forth, like all of you are doing in India right now. Thank you very much. Thank you very much.
Thank you so much, Dr. Shrikant. We had a fantastic talk. It was, I think it must have been very helpful for young students and researchers. as you touched upon very fundamentals like magnetic anisotropy, and also talked about how do we deliver them to the desired sites. Uh, but still, I can see there are a lot of messages coming up in chat box. So these are questions from students and researchers, which who are listening to you, and some of
Okay.
them I can just pick for you.
Would you be able
Is
to
there
moderate,
any
pick a
disadvantage of
few
using this?
questions?
Yeah, yeah, I can. There's question by Lipsa Mishra. Is there any disadvantage of using these nanoparticles in
Disadvantage
MRI?
of
When we use them for MRI, is there any disadvantage of using these nanoparticles?
For MRI, mostly there are many other kinds of
Yeah,
nanoparticles used, gadolinium being one of them, right? So
yeah,
essentially, as I said, nanoparticles, as long as you're careful about the selection of size, it should not be too small for the reason that I mentioned, that you increase the chance of blood-brain barrier, okay? Penetration of that. So one has to work with the right size of nanoparticles and also small volume, small quantity, obviously, which is below the toxic level, and then it should be fine.
okay. And so, what is the appropriate shape and size that is useful for such therapy and imaging techniques?
Yeah. I
That's
showed you the
another question.
influence of the shape and size directly as it comes to hypothermia, right? Clearly, having something beyond spherical, something like cubes, rods, is more advantageous for this. However, one should also think about the biochemistry. Is it easier to functionalize rods compared to spheres? So it's always a question of trade-off, like which one is better for a certain application. But as far as the what we did is to show that you can actually change the anisotropy and get orders of magnitude increase in the efficiency of these particles. So if you want to, ideally, if you want to ask what is the best shape, I would say something that is rod-like, but also has exchange anisotropy in the shell, that it's a core shell rod, which is a little challenging to make. Okay, so a rod-shaped cylindrical particle, which is also core shell, so that you can have the exchange anisotropy on top of the shape anisotropy. They can combine and give you the most effective way. That
Yeah.
would be my suggestion.
We have some more questions are there. Uh, how do you ensure delivery of nanoparticles to a particular target area? That's a
Yes.
question by Jaganya Tripathi.
Excellent question.
Yeah,
Like I said, this is the reason why I mainly focused on the materials part. Towards the end, I mentioned that you do need to do a lot of surface functionalization. So to select specific cells, there are ways of doing it. You can have different ligands, you can have aptamers, you can have antibodies that only react with those specific targets. So there's a way of doing this, and this is of course a very mature field where you choose the right ligand and the right combination of surface chemistry, so that these particles, of course, there's going to be some particles going to other areas, but they largely go to the area that you want them to stay. The other thing is local injection, right? You don't inject it, if you want to treat
yeah,
somewhere near the kidney, you don't inject near the shoulder. You try to keep it local and then use the veins and the arteries that supply, the capillaries that supply the region, blood to that region for transport. So it's a combination of chemical and physical techniques to be able to target them.
right. There's what is the mode of activation of magnetic nanoparticles after
Let me
internalization
stop
into our body?
sharing the screen just in case.
That's okay. That's a question by Chandana Mohanty.
Right.
Yeah.
Can you repeat the question again, please?
Yeah, the question is, what is the mode of activation of magnetic nanoparticles after internalization into our body? Will it be able to kill those cancer cells? Okay, you have
The mode of
cancer
activation.
stem cells. Okay.
The mode of
Yeah.
activation is
You can see the last message.
where I'm actually trying to find where there are 22 messages. Okay, which one? So basically,
the last one that I can see here.
what is the mode of activation of magnetic nanoparticles after internalization
Yeah.
into the body? Will it be able to kill the cancer stem cells? The mode of activation is basically the particles themselves are super paramagnetic. In other words, they are magnetized only when you apply a field, right? That's why we use nanoparticles. Otherwise, you can use any kind of magnetic material. However, we don't want the coercivity. When you remove the magnetic field, they should be demagnetized. So this is why we use nanoparticles. So whenever the magnetic field is there, the particle will be activated. And because we are going to use an AC field, a high frequency, the particle will be activated and it'll have a large coercivity. So that's why I made the distinction between AC coercivity and DC field, because DC field, there will be no coercivity when they are super paramagnetic particles.
Yeah, very nice talk, lot of impression. I think your former colleague Prasanna is trying to ask you a question. I have unmuted him. If Prasanna, can you hear me?
Yeah, I can see Prasanna Sahu. Hi. I mean, I can see that he wrote a message.
Okay, if he is not listening to me, I'll just read his question. Of course, he says that he spent a couple of years at the same department, but never had a chance to listen to your talk. But the question is how to remove the nanoparticles after treatment. Otherwise, it may affect other healthy cells.
Well, that's pretty much the reason why FDA is not giving us approval, right? Because there's no clear answer to that. One has to do a lot more. It's like a chicken and egg story. You know, to get answers, we actually need to do clinical trials with human patients. I mean, a lot of work that we also did deals with rat models, injecting into the rat, we can find out the efficiency. However, the human body is completely different. So unless we do experiments with human patients, more and more experiments, it's just starting now. So unless we do that, we don't have an answer to questions like these. And this is the reason why FDA sometimes takes a lot of time before approval is given for a certain technique. So nanoparticles inside the human body is indeed a problem, right? So we need to take it out, flush it out. Ideally, and again, this is a challenge to all of you students, if you can come up with biodegradable nanoparticles that are magnetic, problem solved. You don't need to worry about flushing them out. However, otherwise, you have to think of ways of flushing them out. But keep in mind that it's not completely bad. For example, there are organs in the human body, like your kidney, your spleen. Their role is to screen junk, right? So you can get basically your spleen, your liver, and your kidney. Their role is to collect junk, all the junk, and like a filter. Okay, so you have a natural filter. So it's okay if some of the particles end up in your kidneys. It's not something. So it's a trade-off between what is more important for you, killing the cancer or living with some small issues in the kidney. And we know that we can survive without even one kidney, right? So essentially, you have to do a trade-off of what is most effective here and what is needed. So, but that is a big challenge. We don't have an answer to that. I also ask you to look at the time because I think we're going past the usual, schedule here.
Okay.
So it's up to you.
So thank you so much, sir. Yeah. Thank you so much, Dr. Shrikant.
If there are any more questions, they can reach me by email, and I'll be more than happy to provide answers.
Definitely.
Okay?
So,
can I ask one question?
Yes, sir.
Yes.
Very nice presentation, sir. And you are giving the complete overview of the nanoparticles. My concern is this in pandemic situation of COVID-19. What is your opinion? Which nanoparticle play a very important role in destruction of, suppose we are taking the mask, but still there is no clear-cut nanoparticle which is going to be safer side for the human to have some mask. So can you give some comment on this COVID-19? Some nanoparticles.
Okay, the COVID-19, again, it's a different kind of protein. As you know, there are two proteins for the COVID-19. I mean, people know these cartoons by now, the spikes, the spike protein. So the spike protein has to be tagged, okay? So that's basically what is needed. So there's some work going on where nanomaterials are being proposed to basically attach to the COVID-19 virus. This viruses are fairly big, so it's easy to target them and attach. So when you do that, you can effectively try to either kill the virus directly or use other ways of taking them away, flushing them out. So there's some study going on. Again, it's very early stages at this point, I would say. In our university, there are several groups in our College of Medicine that are involved in COVID research. Like many institutions, there's a separate COVID program now for research program. We can apply for grants with any ideas. So some ideas are being explored. As far as masks that you said, right now, I would suggest that most of the people, it is okay for using just a regular cloth mask. You don't need to get KN95 masks for regular daily use. So in principle, I mean, I travel to India twice a year almost, and I go to many cities. People in Delhi use masks all the time. So, that's the kind of masks you need, just regular masks.
So thank you very much, sir. On behalf of our university and on behalf of our honorable funder, sirs, on behalf of our School of Applied Sciences, on behalf of our organizing committee, and on behalf of me, very warm welcome and thank you very much. And definitely the support what I get from your email, and now we're talking virtual mode, but I feel that you are very near to me. Why not thinking this is virtual? You have made your talk really physically and more excited. Me also, as well, all the audience definitely.
Thank you.
Nanoparticle will make us some new history in the world to the pandemic situations, sir. And definitely, as you are the editor of a general aligned compound, in future program, we can request you so that next program we can have some paper publication in general aligned compound, so more young people can join with us in the program and have a good means.
Thank you. Yeah.
Thank you, sir.
And thanks for inviting me. And it's great meeting you all online. And I look forward to eventually visiting Bhubaneswar again and coming to your institute in person. All right.
Yes, and definitely after COVID-19, when you are planning to come India, just drop a mail, or if possible, we can call you and have visit our KIIT University, sir.
Definitely. Yeah. Thank you very much.
Thank you, sir. Very, very excellent presentation.
Okay.
Thank you. Bye-bye. Bye, everyone.
So we have in this session, the next speaker is Dr. Ashok Kumar, who is a principal scientist at NPL, National Physical Lab, Delhi, at the moment. And he has been visiting fellow at various places like University of Cambridge, Virginia Tech, USA, and he has been previously he was also at Central University of Chhattisgarh as assistant professor. He has worked over there. He has a lot of publications, around more than 200 publications, and with and several awards. His publications include high impact journals such as Nature Communications, Advanced Materials, Physical Review B, and many more like that. His research interest is on working on ferroelectrics, and so here he's also an associate editor of Science Letters journal, and Current Nanoscience, associate editor of Frontier of Materials. So these are so Professor Ashok Kumar is he's going to talk about this thing. Is it there? Is Professor Ashok Kumar is here?
Yeah.
Okay, yeah. Okay, welcome to AFM 2020, sir. So we are eager to listen to your talk. That is on some biological application of some these functional materials. So kindly, yes, sir. Please go ahead.
Yeah. So good morning, all of you, and I'd like to thank Dr. Ania for nice introduction, and also I'd like to thank Dr. SKS Parasher for giving me this opportunity to talk with you people. So I have already discussed about the last 26th. I had discussed about the how you can fabricate piezoelectric sensors as well as pyroelectric sensors and many others sensor applications. And today I'll be talking about the how this, you know, we all are suffering from this hypertension. India is one of the country where 60% or you can say more than 50% suffering from hypertension or you can say blood pressure. So it is so today I thought, why don't we discuss about the discovery of this blood pressure first. So how it was discovered, and then uh, how you can measure the blood pressure of the human being. And then I'll be talking about what are the equipment that will calibrate or make you traceable or that will indicate your measurement is correct or not using the NIBP simulators. So all these things I'll discuss, although completely, you know, this is quite different aspect of life, but very, very important for the, you know, everyone. So I'll share my slides right now, and then we can have, okay, so and everyone is now, my PPT is visible to everyone.
Yes, yes, we can see.
Oh, okay, so
It is visible.
so today I'll be talking about the basic understanding of the blood pressure measurement devices, and also I'll be talking about how you calibrate your blood pressure measuring devices, because most of the measuring devices are faulty in nature. Even there are many cases where people are having not hypertension or high blood pressure, but due to the wrong measurement people started or getting treatment of high blood pressure. So this is equally important. You should have the proper and correct measuring blood pressure measuring devices. So as usual, again the same problem. Yesterday, I don't know what to do. There is some problem while sharing this. I don't know. Let me have a look.
अशोक सर, आप भेज दीजिए मेल में, हम शेयर कर देंगे यहाँ से।
No,
मेल में
don't
भेज
worry. It
दीजिए।
is yesterday. That day also it happens. I don't know why it is happening. I'm just sharing this. This is fine. Huh? Then share all these.
Otherwise, you
If
can
there is any
send to me.
problem, then I'll.
Yes.
So, okay, fine. So there was it was static. That's why, because so it is my duty to discuss about the institution where I am, and the last day also I talked about NPL. NPL is the custodian of all the even SI units. So in India, we are also known as National Meteorological Institute, means the SI units nowadays based on the fundamental constants. So this is our duty to make it traceable, or device which can measure the fundamental constant with the least or least uncertainty, or you can say high accuracy. So our forefathers are, you know, everybody known, he is known to everyone, Dr. K.S. Krishnan, he's a fellow of Royal Society, and he plays significant role in the discovery of Raman effect, magnetic anisotropy, and he was first director of NPL. And even the NPL having, you know, conceptualized in 1943 and by the governing of the Council of Scientific and Industrial Research. So anyone can go the Google, put my name Ashok 553, and you will get all the detail what I am doing. So I don't have to discuss all those things. Already Dr. Amia has discussed about or
told what I am doing. So, but I am taking care of many aspects of the material science as well as the device application, and that's why today I'll be talking about the human blood pressure. You know what is human blood pressure? So, what is vacuum and how it discovered and what is the history behind the human blood pressure? Today I'll be talking about all those things. In terms of the pressure, look, everybody is knowing what is your blood pressure? 72 120. What is this 72 and 120? This is in millimeter of mercury, mmHg. And in other terms, if you are talking about kilopascal, it is in the range of, you can say, 5 kilopascal or 10 kilopascal to 20 kilopascal. This is the blood pressure range, and this range is you can say far below the atmospheric pressure. But how you measure the blood pressure? This cannot be absolute because you have to measure with respect to atmosphere. So this is always a gauge mode pressure. So in normal human being, this is like, you know, the normal human blood pressure in terms of mmHg is 70 to 120 or 80 120. Similarly here your 12 kPa to 15 or 16 kPa. This is the pressure range which our body is having this blood pressure. Beyond this, I'll talk when it is hypertension, low blood pressure, high blood pressure, or many other aspects of that. So, as I discussed in the first slides, I'll be talking about the discovery of this blood pressure. But before the discovery of pressure, I would like to emphasize the discovery of vacuum. I would like to where from it came from. If you go to the Hindu mythology, Nirvat was, you know, its general meanings is the dispirited, Nirvata. Nirvata means it is derived from the, you know, Nirvat or a Nirvana. This is a whole Buddha, you know, called Nirvana. So before even the Rig Veda, the word is called Anida Avantam, which is not blowing, means it is very near to the meaning of the Nirvatam. It is, you can say, as you know, Rig Veda period is far beyond the BC, like 2000 BC. So similarly, in the early text mention Udava, blow out or go out, or you can say it is you are blowing out. So these are the words which I can found in our, you know, Rig Vedas, Vedas, or, you know, Buddha's philosophy is completely based on the Nirvana. Means it is coming from Nirvat. Nirvat is nothing but the vacuum. So the word vacuum is existing in Indian philosophy long back, you know, modern science. So I'll be talking about how this early evidence of this particular vacuum. So what it means? It is a palpation of the pulse. You know, pulse when the human being, you know, you can say the origin or existence, originate human being. From that time, if you check your, the pulse was there. So the Egyptian mythology or Egyptian philosopher, Gispen, in even the 2920 BC to 52 BC, is conventional called Egyptian chronology, and it's known as the pulse or you know, palpation. But the after, you know, near to the BC, Arastu or Astoy is, you know, Arastu is one of the well-known philosopher all over the world, and the modern, you know, modern scientists come philosopher, but they are experimentalists. People have looked. If you go back to the slide, this Torchley, Pascal, Galileo, Medici, these are the people who are real experimentalists and discoverer of this particular pressure and vacuum. So what was the Aristotle philosophy related to the vacuum? So the philosophy was, Aristotelian philosophy was based on there is no existence of the vacuum, and nobody there in that time to tell, you know, the Aristotelian philosophy is wrong. So you need a very, very accurate or experimental evidence in order to discard the philosophy of Estonian philosophy. So it was not easy for the scientist or the philosopher of that era which can tell, okay, your philosophy is wrong. So what was the Aristotelian philosophy was there is no existence of vacuum. However, even the Indian mythology as well as the Western mythology or Egyptian mythology, there was mention about the vacuum things. But it was never easy for anyone to discard the philosophy of Aristotle. So in 17th century, there were mechanical philosophy or experimental philosophy who firmly said no, vacuum exists, and they showed it. Means air having the pressure, air having the existence in the nature. So this Battista Bellani, who was the first person who measured the water column, and then they told that this is the water column can exist up to over to the 34 feet. So I'll tell you how they have done this. Everybody is knowing about the barometer. So how you measure the water column? You take column inside the water, big bucket of water, and see how far it is going up. So they wrote later, this time these guys are one of the best philosopher in Europe, and this partially later to the Ricci about the who was working on the barometer, and you can see the statement. You can read many books and things. How it was so, because that time also, if somebody is getting something, they used to write their invention or discovery to other scientists in order to confirm it. So they write about, look, what I got, the water column is not rising above or not going above this 34 feet. So the next philosophy come, okay, so let's change the fluid. What does it mean? It means instead of water, use the seawater, use the wine, use the blood pressure, use the mercury. Scientists involved in all these experiments are Ligeti, Galileo, Torsley, Pascal. These were the people who were extensively working on different material, because that time it was not known how and why the height of these particular fluids change with the change in the fluid. So then they performed on mercury. The Vivani performed experiment on the mercury, and what they found, the mercury column only rises 14 times, because wheat is heavier than the water, and it's 14 times. So what they found, the mercury reached 14 times a smaller height than the water. It means water column is 34, then mercury reached only 34 by 14. So you can calculate how much height 2 point something feet it reached. So then it was also easy experiment for making a column. Only you have only 2 feet or 3 feet column, and you can make a barometer. So the concept was proven, air having the weight and vacuum has been established. So it was, now you can understand all those scientists and all those researchers who are fighting against the philosophy of Aristotle, because Aristotle established there is no vacuum. But it took around 200 years, all those scientists, in order to counterpart all those well-established philosophy. So finally vacuum established, and now you can see how it discovered. So then there is a theory of wine water experiment. Then this, Florin Perry Pudy dome experiment. They did experiment on the mountain. They saw the height or change in the mercury column height or change in the other, fluid height is different compared to the sea level. So then it is another. It also depends upon the height. It also depends upon the gravity. It also depends upon the density of the material. So now the theory, all these, and if anyone is interested to understand more understanding on the history of this blood pressure, how, so you must go and read these books like Pascal's The Life of the Genius. Then you can go the physical treatise of the Pascal, the construction of the modern science, Cambridge University Press. All those books you can download and you can read how these things develop with the time. Now let us come to now you understand how we discovered the barometer. Now, once you discovered the barometer, now this will lead to the path of the discovery of the human blood pressure. But before, human blood pressure, that time it was even the 15th or 17th century, it was not easy to measure the blood pressure of the human being. So how people started measuring the blood pressure? As you can see here in this particular slide, this guy is putting a brush or glass pipe tube in the artery of this particular horse. And what they found that 1/6 the diameter of this particular stick was 1/6 inch of the diameter, and 9 brass glass tube was used to measure the blood pressure. What they observed the 8 feet 3 inches blood height above the ventricle of the heart, which rises and fall after each pulse. So from there it was established that this is the blood pressure of this particular horse. So Stephen Hales was well known for their experimental hand, and this will leads you to, or if you read all those things, you will be so excited how these people first try to look. This is the real discovery. They were trying to discover the blood pressure of human being, but you cannot put a rod inside the near to the heart of a human being in that particular days. So they use this brass and the glass tube in the horse, and they found they observed the blood is reaching around the 8 feet and 3 inches during all compression. So in this way it was blood pressure of the animals established. So the another great scientist called Riva Rocky in 1869, everybody, and the protocop sound, which is well known protocop sound, everybody, all human being is knowing. If you go to the hospital, if you have like 20 years back, everybody is using the non-invasive blood measuring devices based on the protocop sound. You can see here there is a Russian surgeon named Nikolai Krotokov, who developed the modern technique using the stethoscope, which is measuring or listening the sound of the blood pressure flowing through the artery. And you can easily say that this is the systolic and diastolic pressure. I'll come to that particular point, what is systolic and diastolic. But this was the guy who developed, Riva along with the Krotokov, the modern equipment which measure the human blood pressure. And can you imagine how was the discovery and how big was his paper? The paper was only 207 words. 207 words, not a abstract like kind of things. And this is the leading last 200 years, you can say, or still it is the backbone of the human blood pressure. So only 200 words paper can change the world. So he discovered the systolic and diastolic pressure. The systolic pressure, the first sound appeared after the fall of the mercury in the manometer, and the heard systolic pressure murmured, which became sound secondary, you can say, called diastolic pressure, and then there is no sound. So anyone, look, if you go anywhere to measure your blood pressure while not, oscillometric method, some normal method, then what you do, you can put the stethoscope in your ear, and then you can easily hear the sound once the first sound appears, because because you are increasing the pressure above the human blood pressure, and once you release the pressure, then the artery will flow, the blood will flow through the artery, and this will give you the first sound, that's called systolic. And where the murmur, after murmuring, the sound will stop, that's called diastolic. So in 1920, this mercury-based sphygmometer, you can see this is the device which was developed by the Philip Midgate. So this was the first device and the first theoretical as well as experimental, this Russian guy and this, Philip Midgate, which developed these blood pressure devices. So now you understand the history, discovery of the blood pressure, discovery of the, vacuum, discovery of the pressure, how it was, it was a long path in order to discovered many aspects. So definitions, like many blood pressure, how it is defined, as you know, force exerted by the blood against the wall of the artery, that's called blood pressure. And the systolic, as you know, it is defined by the once a large artery when the heart is contracted. And once it is the diastolic, means defined as the pressure observed on the large artery when the heart is relaxed. So diastolic once heart is relaxed, systolic once heart is contracted. So hypertension, if it is in the elevation of either diastolic or systolic, it is called hypertension. So here, this is the pulse, you can say. If you I'll give you some, we have done some experiment where we observed this diastolic, and this is the systolic pressure, and this is the mean pressure. So, and how it is happening, there are many ways you can have the measurement. Now modern technique, modern devices, many things. I'll come to the point and I'll talk how it is, how it will work. So it will have look how the blood is flowing through the various arteries and what will be the what are the flowing speed. Like if you know what is the speed of the blood, like 0.5 meter per second to 1 meter per second, and the systolic and large vessels. So this blood pressure, if you have look this high blood pressure chart, the both systolic and diastolic go high. That's the serious condition. Then the systolic hypertension only the when the BP goes high, and the diastolic means the hypertension only diastolic BP will go high. Low blood pressure means both will go down. So you can see here this is the area. Probably I don't know whether you people have. You can Google it and you can find out how this is the varying on the various area and the various type of high blood pressure. It is defined by already and accepted by world community. And this is the area where people having the high blood pressure range. The red one indicates all over. I am giving you a globe chart, how it is distributed globally. And these are the area where blood suffering from the low blood pressure, then the high to low, and how the prevalence of the non-hypertension, newly detected hypertension, pre-hypertension, and I was talking of the globally, and now you can say the Indian context, like 20 to 40% of the urban adults and around 20% of the rural adults are suffering from the hypertension, which big amount of, if our population is 130 million, you can understand how much is we are suffering from. It's a big disease, and due to this hypertension, people are suffering the cardiac arrest, suffering from various type of disease, kidney failure, or you can say many other organs failure. Leads to even cancer also, many other, it can be leads to any disease which is known disease. But hypertension, so what is our role? As I already discussed, this is the diastolic. It will pressurize, and you want to see that the maximum of this will give you the systolic, and then the bottom of this particular, all depends upon, and this graph is related to where you are measuring the pressure. Look, you can see here the left ventricle to arteries to capillaries to venous vein, right, and these having the different. So what is the best range where you can measure the systolic and diastolic pressure? Means artery is the best place where you can have high detection. You can detect the systolic and diastolic pressure, and you can say if the person is suffering from hypertension or not. So there are many methods and technique which is used for the measurement of the blood pressure. So in terms of, if you have, in the very beginning, I talked about how the scientists measure the blood pressure of a horse. That is called invasive catheterization method, means catheterization method. Even right now, if the person having the open heart surgery or bypass surgery or many major surgery, then the doctors want to have the invasive catheterization method, where they want to put the sensor near to the heart in order to understand how the blood is, or how the blood pressure of the human or that particular or subject having changing with the during their operation. So invasive catheterization method was in the very beginning, but still for the sophisticated operation, scientists or doctors are using this. And then the auscultatory method, which is, as you know, normal, based on the mercury based blood pressure measuring devices, or you can say manual devices. It's not based on the oscillometric. I'll talk about the how oscillometric, nowadays everybody having the automatic blood pressure measuring devices, how it is working. And then there are many other techniques like infrared based, infrasound techniques, ultrasound techniques, impedance techniques, arterial tonography technique, ovulatory blood pressure monitoring technique. There are many techniques. I cannot cover all the techniques right now in this 40 minutes of Spain, but you can, if you are interested, you can go. So this is the some of the examples where how ultrasonic method is using to measure the blood pressure. You can see here, you can put certain, they generate certain, piezoelectric transducer. It generates some sort of the ultrasonic waves. It will pass through the, your artery, and it will detect the change in the blood pressure. That's it. So that will give you more highly accurate systolic and diastolic pressure after, all kind of electronics and filter, using the filter, amplification or other process. But this is the technique, oscillometric technique, which is right now used all over world. And most of the devices, which is everybody household devices, based on this oscillometric technique. What is this technique? This is technique, you generate a pressure above than the, just you can say, the blood pressure of that particular subject or that particular human being. And then once you release the blood pressure, human heart will generate a pulse. And the pulse, you can, if you filter that pulse from the during the decreasing pressure, then that, if you do the Fourier transform, you will get this kind of sinusoidal or pulse type of waves. That's called oscillometric methods. And 2/3 mean artery pressure is 2/3 of the diastolic pressure, 1/3 of the systolic pressure. And with this, there is a common convention worldwide. 0.7% of the mean artery pressure and 0.5% of mean artery pressure will give you the systolic and diastolic pressure. So this is after many clinical trial, scientists and doctors has come to the point, this is a very good technique, but then they used to develop some algorithm in order to filter these particular oscillometric pulses, and come to the conclusion that this much of mean pressure will give you the systolic and diastolic within certain limitation. I'll come to those points also. There are tonometry. You can have many methods. It's very beginning. You can see this is the first sphygmograph, which was designed by the Julie Mary in 1860. And after that, now techniques developed and many things based on this tonometry. They also based on the measure the pulse pressure and converted that pulse into the systolic and diastolic pressure. Nowadays, now you don't need even the oscillometric based blood pressure devices. you have a watch, a smart watch on your hand, and that will give you the blood pressure of your body. So how it works? It works based on the wrist, having the sensor sitting at the bottom of the wrist, a watch, and you have the median nerves kind this, ulnar bones here, and you have the sensor radial artery, which sense the change in pressure or change in pulse by sensing like this. So you have the sensor plunger, and we have the radius bone, and this will generate, this will detect the things, filter it out, do the calculation, and measure the systolic. And because all this based on the human blood pressure pulse, how the pulse is behaving. This sensor is sensitive enough to detect that change in pressure of the radial arteries. That's it. Here you have the cuff arteries where you want to measure the pressure. Similarly, we have the wireless wristwatch, and you don't have anything right now. The similar kind of things. Now you can understand. Earlier there was some sort of extender cuff, but this kind of entirely wrapped cuff is completely 360-degree, and where how you mount it hardly matter, and that will give you a very accurate blood pressure while you are running, you are playing, you are doing an exercise, during the healthcare. And these kind of devices is the boon for the blood pressure or measuring online or in situ. Omron has already launched this in 2017 in the market, and many more other new stuff came to the market. I'll discuss some of the new stuff, which will oximeter, which is nowadays very useful for the COVID patient, which will give you the oxygen level of your body, and at a time same time it is integrated with your smart cell phone. So this will give you a kind of blood pressure or your presence of how much oxygen level in your body. So you don't have to everything is going on single in your pocket. You can save it for another evaluation for that your physical condition. You can share with your doctor, whatever be. So these are the things has been developed already in the market. And then I'll come to the point where we have also trying to develop some on low cost blood pressure measuring devices. And this is the devices where you can have a small cord, and then you have the small electric circuit with the microprocessor and things, and that can measure the blood pressure using your smartwatch or a smartphone or smart devices, which is always with you, and it doesn't require too much cost. So you can develop in your lab having your low cost blood pressure measuring devices. As I told you, what is the direct blood pressure measurement sensor? There are many invasive techniques where you can measure the. I don't know how much time I have. Okay. How much time I have? Can. Hello. How much time I have? Can you, Chairman, can you tell me how much time I have right now?
Hello.
Hello. Yes.
Yes, sir.
How much time I have now?
10 minutes.
Okay. Fine. So, this is the way
Yes,
you
sir.
can have.
Yes.
So now, like right now, if you have, I don't know how many of you are in all the sessions from last 2-3 days. I was also talking about we are trying to develop some sort of the fiber fabrics which can detect all your physiological changes in your body. So this is the smart clothing which integrate health and the maximum comfort, which is based on the nanosecond pulse near field sensing technology. So what does it means? It can measure the mood of the human being. Suppose that if you have a fight with your wife, that will tell you that now you are in mental stress. So be careful. If you have a good mood, like something, okay, you are now getting overexcited and all the things, the blood pressure is changing, that will give you the information whether you have some distress or some sort of the discomfort or some sort of the pleasure or some sort of, so that will give you all kind of physiological information based on this nanosecond pulse near field sensing technology. So a textile or a kind of clothes, t-shirts or pant, that will give you all kind of your physiological change. So this is happening due to all the advancement in the science. So let's come to the point where I want to discuss about, I don't know where is those my slides. Okay, I think, so what I'm talking about is how we will measure this non-invasive blood pressure instrument. So first, in order to, for test proof that this device is working fine, we have to do the test for the non-invasive blood pressure instrument, get air leakage, how it is the leakage as per the OML guideline R16.2. So air leakage, a rapid exhaust time. So what I am trying to, as I told you in the beginning, the measurement of wrong measurement of the blood pressure is one of the major cause for the hypertension. Even the people are not having the hypertension or high blood pressure, they are treated with the medication. So that is, should not be so. As per protocol, all medical devices should be calibrated or tested within 6 months or in a year with the proper testing procedure. So these are the proper testing procedure which is required for a non-invasive blood pressure instrument: air leakage, rapid exhaust time, condition for the cuff, tube, bulb fitting, scale visibility, contamination of the glass tube and mercury, cuff inflation, deflation, control security on of the mercury spill out. All those things we have to find out. And then you can see mercury sphygmometer should have a fixed like immovable object. Calibration interval is 36 months, check interval 6 months. These are the protocol already accepted by various physicians based on the clinical trial or worldwide. So recommended sphygmometer calibration and check intervals. So this is the way you can have a traceability of air. So if you, these are the mercury free, dial gauge, oscillatory based digital non-invasive blood pressure devices. So this should be have the digital devices, then simulator. I'll talk. We have established this simulator, pulse generator, and all this based on this, like our, since we are National Meteorology Lab or Legal Meteorology Lab, we need to have these two type of devices in order to calibrate all these digital NIBP devices and mercury fit for the common people. These are the simulator which is already in the market. So what we have done, you can see here the another NMI Criscoria who has set up this particular non-invasive measurement for the how you can taste or measure the their, this is they have published in the, and this is the setup for the a simulator. So what is the simulator? It generates the human plots based on some logic and the data given it to based on the clinical trial. This is a huge task. It's not easy to have. So this has servo motor or sometime another kind of pulse motor which generate pulse and other things to have this servo motor, pulse generator, pulse transducer, data acquisition board, and the automatic blood pressure monitoring devices having the cup. I'll give you that will be more this is more clear, and you can have look how it is calibrated. So this is the NIBP monitoring devices. Here it is, which is giving like 120/80. So this is the digital devices available in the market, hospitals, household, personal human beings which are suffering from hypertension. So how it will be tested, whether the value given by this particular device is right or wrong. So it generates a matching frequency or pulse near to the same systolic and diastolic pressure, and then we measure how much difference between these two, and this is calibrated, by the way. So once you have the pulse generation is calibrated, and this is dynamically calibrated with the devices, you can tell ki this device is working fine or not within the limit. So this is our setup in CSI or NPL, where we have developed a NIBP simulator system for the performance validation of NIBP devices, non-invasive mode, you can say. So here it is from the Fluke company, and this is the Omron NIBP devices. We have developed some algorithm or some set of the like blood pressure, like 80, 120, 70, 120, 60, 180, 100, 200, whatever very high tension. And then we tested whether this device is giving as per this simulated value or not, because this is calibrated. So once it is calibrated and it is matching with the value within the limit of plus minus 3 mmHg, we can pass it to this particular device is passed in our test. And this is very good for the human being. It can be used anywhere without any obstacle, without any problem, and this will always give you a correct, unless and until blood pressure is also based on the physiological changes. If your mood is not right, you have the another blood pressure. If you are having some certain different environmental circumstances, blood pressure will be different. But as usual, you have to be cool, calm, your arms should be in the rest position and having other, and then this will give you a perfect value. And we have also developed some high pressure sensor, which is also useful for physiological application, blood pressure sensor. And as I told you, this is from 10 kilopascal to 50 or 100 kilopascal. This having the very high sensitivity. This can detect the blood pressure very easily. We are trying to develop electronics in order to have the low cost blood pressure measurement devices. Blood pressure also. So this is you can say already developed, published, and we are also working on many other devices which cannot be included in this short period of time, where we have sense very small pressure, and which can have many human either the cornea pressure, or blood pressure, or your pulse rate, your heartbeat, many things we can be converted. Since these sensors are very good, they can sense and transfer from this to electronics, and that can be visible, that can sense all kind of values. So this is another example where we have the low pressure, high pressure, and the sensitivity is very high, and we are ready to have developed some sensor with the proper electronics, which can convert this capacitance into the voltage, and then voltage in the digital display devices, digital display unit. So all these are underway. This is the uncertainty evaluation, and I think this is the another simulator which is used by Chris Korea, another NMI, and they have developed the pulses. As I told you, this is the uncertainty performance, and this is very important because they have use many pulses from the 1, 2, 3, 4, 5, 6, 7, and based on that, based on the uncertainty, they calculate the uncertainty for the pulse amplitude, uncertainty for the cup pressure, systolic and diastolic pressure, and which is very much needed. So India is also doing good in this regard. We have developed, set up a NIBP simulator in order to calibrate, test it, or type approval for any kind of non-invasive blood pressure devices. This is the one of the devices, and there are
Sorry to interrupt you, sir.
Yes.
But we are running late.
Yeah. Now I'll be finished within 1 minute. Okay. So let's. This is the last slide. So right now we have many other devices. You can see here the latest path. There is some silicon-based devices where electron mean free path in the atmosphere, and which is used this as a vacuum channel, and this will be also having a good devices in terms of the future technology. So let me conclude now, because so what we want to discuss here is how the blood pressure is discovered, how the new devices came into the market, what is the background or basis of the new devices, oscillometric method, oscillatory methods, another process normal, and how it is calibrated or type approval can be done. What is the proper condition of Indian as NMI? We are at par with any country in the world. We can have the taste and type approval of any kind of blood pressure measuring devices. Thank you very much. Yes.
Okay. Thank you so much, sir, for the nice presentation. So I have a question.
Yes.
What are the kind of nanomaterials that can be used for developing such non-invasive blood pressure systems?
Look, anything which can really sense the change in the blood pressure, like if you have in both kind of things, like resistive, if I'm talking about the resistive sensor or capacitive sensor, if you have change in the resistance due to the change in your blood pressure or your heart rate or your pulses, that can be. But the thing is that your nanomaterial or nano device should have such kind of sensitivity which can amplify and can give you the better result, because anyway this is a very small pressure that should be
Yes, yes.
sensed by that particular device, and that can be converted into a real display unit. So this is the only fact that one should keep in mind for device development. You have developed these devices, but the sensitivity should be high in order to detect the pressure of 10 kilopascal, 1 kilopascal, 5 kilopascal, very small pressure. Yes.
Do you have also developing system for the eyes? There's pressure eye. There's also
Yes,
concept of
yes,
eye pressure.
that's called tonometer. We are working on that because the range of eye pressure for glaucoma and other things around 10 kilopascal or 1 kilopascal, 5 kilopascal, very small pressure. So same thing, you have the device like we are working on. We have initiated one project based on the graphene oxide or reduced graphene oxide that can be easily mounted to the in the cornea, having lens, which lens you are making, a plastic
Yes.
lens. If you will mount on that, you can easily measure the pressure inside your eye, and that will save the life of a person which is suffering from glaucoma, that can go many other further diseases. So that's we are also working, and this is the same pressure range. So that's I tell you, this is a good area where it's not a you need many things, but of course related to eye you should have careful the your material should intoxic, you should have a good lithography and other things for the fabrication of the devices which can mount in the lens that can put inside your eye. Yes.
Okay. So in the chat box, I don't see any question. Okay, there are no more questions from the participants. So I thank you, sir, straight away.
Thank you, all
Thank you
of
so
you.
much, sir.
Thanks. You also, you have wonderful, and I would also like to thank Dr. Parashar, which is giving opportunity, all the KIIT community. I am always, from it is always near to my heart. So that's thank you all for giving me
Sure, sir.
opportunity. Thank you.
So thank you, Ashokji. A very
Thank you.
nice presentation. You have teach many things that we don't know actually. And your talk will definitely given a very insight of the different pressures and the complete ancient. Basically, all the science is from the ancient, but we are forgetting all those technology. So thank you very much. Enlightened us about the whole devices, and on behalf of our KIIT University, on behalf of your founder, on behalf of Applied Sciences and on behalf of organizing committee and on behalf, heartily welcome and many thank you for accepting and giving the wonderful talk, the pre-workshop conference and pre-conference workshop as well as this today. So thank you very much. And we'll discuss in the future and see how NPL and KIIT University can do some wonderful things in the pandemic situation of COVID-19. Thank you. Thank you very much. Namaskar.
So I thank Dr. Ashok Kumar and Dr. Amiya Priyam for chairing the session and Dr. Ashok Kumar for giving a wonderful talk. Now the next session will start. That will be chaired by Dr. Shweta Padma Praharaj, Associate Professor, Department of Physics, School of Applied Sciences, KIIT University. Dr. Shweta Padma, please take over the session.
Thank you, Bhavya. Good morning, everyone. So for this session, the first speaker is Dr. Rajendra Kumar Sharma. He is the chairman and MD of Spell Technologies Private Limited, Pune, as well as he is the MD and CTO of Surya Power Farad Energies Limited, Pune. Along with that, he is the partner of Center of Excellence on Rechargeable Battery Technology, a mighty Government of India initiative at Center for Materials for Electronics Technology, Pune. Now, he has been in the field of energy storage and capacitor manufacturing since 1986, and he also owns the India's first and only supercapacitor manufacturing facility. Now, he deals with products like capacitors, supercapacitors, lithium-ion batteries, sodium-ion batteries, aluminum-ion batteries, and he provides some integration and system supports for PF corrections, harmonic mitigation, renewable energy, and electric mobility. Now, he works on materials like activated carbon, graphene, activated materials for batteries, etc. He also deals with special purpose machine development for these energy storage devices like capacitors, supercapacitors, ion batteries. Now, among some of his achievements, in fact, he has been the India's first in many of the achievements, some of which I'll highlight are the ultra low ESR plastic film capacitor, a supercapacitor and its manufacturing setup, Argeo-based high-performance supercapacitor, in-process development of lithium-ion, sodium-ion, and aluminum-ion batteries, indigenous supercapacitors, based electric two-wheeler and three-wheeler vehicles, indigenous, he supports the development of 2 electric buses also, that is lithium-ion battery-based. Among some of the awards, he owns the Vijayashree Award, Rashtriya Udyog Ratna Award, Times Quality Brand Award, Entrepreneur of the Year Award, India 5000 Best MEME Award, India Achievers Award for Quality Excellence, Achievers World Leadership Award from Moscow, Rashtriya Nirman Ratna Award, and etc. And even Prime Minister has appreciated his work on Argeo-based supercapacitors. Now, among the affiliations and engagements, he is affiliated to National Mission for Electric Mobility, Mission Innovation, DBT DST, that is for high-level stakeholder, panel member of DST's Electric Vehicle Energy Storage Policy Drafting, Indian Science Congress. He is a permanent member of that. He has been in the board of studies for 9 educational institutes and universities, and also in the syllabus committee of some other government autonomous universities. Apart from that, he has published some papers and also filed almost 14 patents. So he has a very long profile, and I welcome you, sir. And he is going to give a very exciting talk on the energy storage, past and future. I hope everyone will enjoy the talk. So without wasting much time, I welcome you, sir.
Thank you. Thank you, Dr. Shweta. And good morning, everybody. So without wasting time, I'll just start because we're already late. I think I will switch off my video because some bandwidth issue is also there. I'll just switch off the video first. Okay, fine. Yeah, Dr. Shweta, are you able to see the screen? Could somebody confirm the?
Yes, sir, it's visible.
Yeah,
You
yeah.
can continue.
Okay. Thank you so much. Yeah, basically on behalf of KIIT and my company, I welcome you all, and I will just briefly go through our company's profile, what we are doing in a fast forward mode. We are into capacitor manufacturing. These are fan capacitors, motor lighting fixtures. This is power factor correction and APFC panel application capacitors. This is smart KVR panels. This particular capacitors are used for in electronics UPS and power electronics part. These are high tension capacitors. We manufacture up to 3000 volt for AC application. And coming on to the other part, we are into super capacitors also. And we are rated among world's top 10 companies. On your screen, you can see the number of companies which are from either US, Europe, Korea, Japan, or Australia. We are rated among the top 10 companies for the super capacitor in the world. And briefly, I will go through the product line of super capacitors. This one, 3000 farad and some module for general purpose application. This particular module is like for energy harvesting, you for braking energy capturing or some regenerative energy like at the docks or some shipping yards, which can be captured into this. This particular module was developed for solar application. This is again power backup support, like for UPS or not exactly UPS, but for critical applications like in nuclear power plant or railway signals like that. This particular thin supercapacitor is for controlling the pitch of the windmill. So this is a module. I will discuss it in the subsequent slides. This is a module of battery and supercapacitor combined. Now, apart from this, we are partners for Center of Excellence with India government for creating an infrastructure of indigenous lithium-ion batteries, especially to support enterprising students like you, who are the student, or some enterprising companies who want to start indigenous manufacturing of lithium-ion batteries. So in this case, our deliverable is practically startups. To be honest enough, not batteries. Batteries is a byproduct. This particular is again a center of excellency, which has been recently sanctioned for 3D printing of the battery. In fact, we'll be developing the technology for printing of batteries in 3D and batteries and supercapacitors, and also a memoristor. A memoristor for youngsters like you, some technology savvy people I will just give you a small brief of memristor. Then you can Google it. What is that? Memristor is basically a future device, and it is a combination of resistance, capacitor, and inductor at nano level. And basically, it is the core component for artificial intelligence. The Google Sophia, everybody knows about that. That particular thing for artificial intelligence, memristor is the vital component. With the memristor in practical life, your computer will not require software. Your computer will not require booting. It is like that. That is a future device. So we have taken the task to develop indigenous memristor. This is with the help of nanotechnology. That itself is a big topic. So we'll just go through via industrial collaborator to many research institutes of India. Basically, you will get confused by whether we are industry or academic part. I would like to clarify on that part. Basically, we are an industry, and normally, if I tell you, a normal business goes by B2B part, business to business. But over a period of time, what has happened? The mode of business have changed, and we have not taken a note of that. Like China is typically operating on C2C, not B2B. Now, what is that C2C? Of course, it is copy to China. So whatever products are available worldwide, there is 99% chance the copy of that product do exist in China. And that is the backbone of the economy of China, that is C2C. Now, India is moving fast forward on that technological part or some Atmanirbhar part. And we want to become a world's leading economy. But I would rather suggest the model for India is not B2B or C2C. It is A to E, A to A. What is that A to A? I have coined it up. It is Atma Chintan to Atmanirbhar. So definitely our Prime Minister has given a slogan, and we are working on that part, which is Atmanirbhar Bharat. But Atmanirbhar Bharat cannot become unless and until we do Atma Chintan. And for that Atma Chintan, there has to be a very big focus on technology, and there is a very big role of the universities like KIIT and other universities and the research institute and the industry. So that is a trial, because I call it technology. Over a period of time, technology has become romantic. Every morning you get up and you have something. So every day, that first thing that we can synchronize only. We have a close association between the industry, the university, and the research institute bracket government of India. Because government of India will be representing in form of a research institute, not physically any ministry or like that. So that is a typical Bollywood style equation wherein we call it as a love triangle, but technically it is a particular model actually. That model is known as triple helix innovation model. So we need to follow that, and because of that, only we can become Atmanirbhar and A to A part. So now coming on to the today's topic, very hot topic, burning topic, energy storage technology. Because why it is burning, why it is hot? Because if I ask all of you, everybody is very happy with their mobile. The configuration, the hardware part, the software, almost most of the people are happy. But one thing they are not happy about is the battery. Right, because the technology has developed at very high speed, but energy storage technology unfortunately was not able to keep the pace with that development. And today is the scenario, all the power hungry device, because all these devices have become very power hungry, and battery or the energy storage is finding it difficult, finding it hard to honor that. And when it comes to energy, we have only one part in mind most of the time. Okay, energy cannot be created, nor it can be destroyed, but it can be stored. Great. What do you mean by stored? Storage is actually a capturing of available energy at one time for the use to be used later on, and to store that energy, that is a accumulator or a battery. And for your just general knowledge, apart from this, even nature is the first creator of the energy storage. There is a biological energy storage device. Human body, plant. Okay, like in human body, you have an energy storage device, which is liver. Of course, liver can store 5 to 6% of the energy. 5 to 6% of what? Weight of the liver. Okay, and your muscles can also store 1 to 2% of your muscle weight. So that kind of energy storage device is already bestowed upon us by the nature, even in the plant, because human beings store energy in the form of glycogen. Plant, they store in the form of starch. Even the very great part of nature is that uterus in women during the time of pregnancy, that uterus gets converted to energy storage device, temporary. So definitely energy storage is a big field, and our focus is purely on the man-made energy storage device. So we'll briefly go through that energy storage device, and that we calculate in terms of what is the capacity and for how much time. So on your screen you can see the x-axis showing as capacity and the release time on the y-axis. The first person or the first thing which comes out to energy is capacitor. Capacitor was invented way back in 1745. So capacitor fall under electrical energy storage device. There is another energy storage device, a coil, a superconducting coil, that also falls on in this particular domain of electric storage. The other is a flywheel, that is a mechanical form of energy storage. The other one is chemical form, which is hydrogen. The other is electrochemical devices. Now this electrochemical part have seen lot of evolution. Redox battery, lead acid battery, lithium ion battery, and so on. The other is a pumped hydro system, energy storage again. The very small, I think many people may not be able to read, I'm sorry for that. The small particle which is seen on the screen is a compressed air. That is also an energy storage device. So in today's topic, because the most happening area of energy storage is electrochemical and electrical. Other part have already reached their theoretical limits, and it is long back they have gone into saturation mode. So we'll discuss on electrochemical and the electrical part. The first thing is a popular device. Electrochemical is a battery. So the present development of the battery, the future part, but I am of the view you cannot go on future without your present, and your present is backed up by your past. So let us briefly go through certain past of the battery. On your screen you will be seeing something written, and I bet most of you must be wondering what is that. Okay, this particular thing is extract from August Samhita. What is August Samhita? It was written way back 5,000 years before, 1550 Samhita, and there is something written by Rishi Agastya, and this particular is available in Agast Samhita, that is at Ujjain. There is it is kept in that original writings are kept. Now, what is translation of that? Like you are wondering, one of the engineer from Pune was also wondering what is that, because he had passed out his engineering in somewhere in 1891, and he had one mission to explore the scientific relation of the old Indian writings of Veda and Agastya Rishi, what has written. So he went to Ujjain in search of that, and he found out that, and he was finding it is something related to electricity, but he was not able to translate. So he took that extract and went to Nagpur University for translation, and that is a translation what you can see on your screen. And this was something very astonishing, and it was given to one of the electrical engineer, Professor Hole. I'm not wrong. Yeah, Professor Hole from Nagpur University at that time. It is in 1891. Okay, and he thought he will make that, collect that apparatus and do something, and that particular pot is visible on your this thing, which was made with the procedure of Agastya Rishi, and this turned out to be a Daniel cell. Of course, Daniel cell was invented in 1836. This is exactly a Daniel cell. So with this, we should appreciate our old Indian part. We have been very forward in that part also, but recognition was not there. That is a different part. So let's move on to the battery portion. And the battery portion has gone into lot of transformation over a period of time. Lead acid battery was there. Volta has done something. Everybody has done. But a French physicist in 1859, Dr. Gaston, came out with the lead acid battery, which is very popular. You can see on your screen that is the internal structure of the lead acid. We'll understand how lead acid functions. On your screen, you can see a fully charged lead acid battery. A lead acid battery consists of anode and a cathode, which is made of a sponge lead, negative active material, and the positive active material, and a separator with acid, which is roughly around 31% added to the water. And now what happens if I connect a load to this? The load has been connected. We have switched on. Look at the reaction, what is going on in that. This particular reaction, we call it as reduction oxidation reaction. And that's why that is how the electricity is produced. And finally, the battery gets discharged. And what is that chemical equation, which is visible on the top of your screen? And earlier there was a H2SO4 visible. Now that has got converted into water. Many people, when you see your battery lead acid of automobile, they call it "Arey pani ho gaya iske andar." They are right. It has got converted to water. Okay. Now when you charge this, again the H2SO4 part comes into picture. So that thing goes on and on. These are rechargeable batteries. This kind of batteries are known as secondary battery. The primary batteries are batteries which cannot be recharged one time use. So these all fall under secondary battery. The most advanced part of the battery, when like I was referring to you, the Atma Chintan and Atmanirbhar Bharat, if you talk about lead acid, every lane almost of a rural India or a urban India at the unorganized level, you will find lead acid battery manufacturer, right? They are making and earning their livelihood. But when it comes to a new technology, which is everybody is advocating, lithium-ion battery, there is no one in India the 70% of the world research on lithium-ion batteries is either done in India or by an Indian. But unfortunately, we don't have a single manufacturer. That is a pity, because technology has developed to such a high speed and we have been left out. I want to cover up that left out part also in my mission, apart from manufacturing supercapacitors and all that. So lithium-ion battery. Now, what is lithium-ion battery? How it differentiates with the lead acid battery? The lead acid battery works on the principle of reduction and oxidation, whereas lithium ion, it works on the principle of intercalation. What is intercalation? On your screen you can see it is a self-explanatory picture. Intercalation, the ions are inserted in the anode when it is charged, and when it is discharged there is a release of ion and there is a flow of electric current at the load level. So that is a simple process which is followed. And when it comes to lithium ion, the family is very large. Every morning you get up, somebody comes in picture, look, I can do the things better. Then it is characterized by either some lithium ion cobalt, lithium ion phosphate, lithium nickel. Lot of permutation and combinations are there. Now the major permutation combination is being displayed on your screen, which is LTO. LTO means lithium titanium oxide, LFP, lithium iron phosphate. Now I would like to highlight on this part. See, all the lithium ion batteries, when you call it as LFP, LMO, NMC, LCO, they are basically known by the cathode of the material. Cathode, because you leave aside this LTO, the other chemistry, the anode is common in all the chemistry. The anode is made out of graphite, and the cathode is either made out of lithium iron phosphate active material, or lithium magnesium oxide, or nickel magnesium cobalt, or lithium cobalt oxide, or nickel cobalt, like that. Okay, now this particular LTO chemistry is not referring to cathode. It is referring to the anode. So you have one family of battery which is known as LTO, wherein the graphite anode is replaced by lithium titanium oxide, and the cathode will be either of this chemistry. Okay, you need not to get confused. It will have either of this chemistry. So I've just formulated this chart. So you have so many options. So everybody is confused, which is good, which is bad. I would rather put up in a way, there is nothing called has a good chemistry or a bad chemistry, or a good battery or a bad battery. It is all as per your application, what you need, what you require. So this hex particular chart, you can visit this chart at your own leisure, because we're already running short of time. So I may not be able to explain all the part in this particular talk. You can, all the picture is self-explanatory. That is also there on our website. So we have analyzed these batteries on different level, like life, like safety, like specific power, like specific energy, cost, and lifespan. Out of this, cost is very important part, because everywhere, it is not India. You may have very hi-fi chemistry, very thing. People asked about the efficiency and the cost, the success in the market, because I have to make the battery and not keep it in my house. For lab level, it is okay to do the things and this thing, but at the end of the day, I have to sell it out. So the cost is very important part. So all the research part is concentrating on the cost and the specific energy density and the power density and the safety. So there are too many parameters, and all these present lithium-ion batteries are having one typical problem, because now I'm coming on to future part. As in my talk, I've already committed the present and the future. So whatever chemistry I've told you about lithium ion, it is the present part. And presently, the problem statement is there. All this chemistry are having liquid electrolyte or a semi-liquid electrolyte, which is known as a gel, which is very popular in your lipo batteries in your mobile. That is a semi-gel. Otherwise, all the other part is having liquid electrolyte. This liquid electrolytes, they have very typical problem, which is displayed on your screen also. These problems are what it is organic solvent. It is a very highly inflammable. So in case of some abuse or some accident, it is a risk of catching fire. Then there is a high resistive area. What is this high resistive area? Any resistive component, when current passes through that or current is involved into that circuit, it gives a Joule effect, a Joule heating effect. So that is why the battery gets hot. This particular part contributes to the heating of the battery. And electrolyte decomposition, of course, it is there. When heating is there, when high voltage is there, there will be a decomposition activity going on. So the thermal runaway is there, and the most important part, the leakage problems are there. So how you resolve this? Now this is the next generation battery. My whole idea of putting up the future part is also to instigate you people. I know there will be many researchers, there will be many students, many aspiring people. So like if you want to do something enterprising, you need to follow 4 rules. One, there should be aspiration, so which I'm trying to induce. The other part will be a desperation, because once you understand that and explore, you will be desperate to do something. Then there will be inspiration. Your teachers, your all around chain, Google also is an inspiration part. And then there will be a perspiration, the hard work. Then the things will get complete. So okay, that is a philosophical part. We come on to the main topic of ours. We should not get diverted. Okay, this solid state battery. What is this solid state battery? In solid state battery, the liquid electrolyte is being replaced by a solid electrolyte, not a gel, a solid. And with this part, what happens? You don't require separator. Okay, like in the lithium-ion battery, you must have seen one separator is there. The separator is required to electrically isolate anode and cathode. Okay, and plus the duty of separator is to electronically isolate this anode cathode and allow the ions flow. But when you are using solid electrolyte, that itself is a electrically isolating material. So you don't require the separator, though your cost has gone down. That is a great part. So now how this works actually. The ionic conductivity is also good in case of this thing. And when I'm saying this is a future part, the question will come onto your mind, is this batteries available commercially? I would rather say yes or no also, because the small part of this people have tried to manufacture. The biggest challenge in this solid state battery is actually a manufacturing. Lab level also the work is going on, but the biggest challenge is at manufacturing level. So it is predicted these batteries will be a reality. Google is predicting around 7 to 10 years. We are also working on the project, and we are predicting around 4 years. So let's see how the things go. So these batteries will be coming up maybe in next 7 to 10 years. What is the benefit of these batteries? Fast charging is possible. Say the tune of 6 times fast charging is possible. The increase in energy density is definitely there. It is 2 times, and the leakage current is very low because of no resistive component going into that. The parallel resistance is not there. The equivalent circuit and all that thing being solid and no inflammatory part, you don't require separator. Okay, now this is the module in front of you which explains everything. Okay, if you see the difference between the module, because a single cell of the battery is hardly 3.7 volt or 3.2 volt or something. So when you want to use it for 12 volt, 24 volt something, you have to assemble n number of cells. In lithium ion, you have to take separate cells and do the external assembly. In supercapacitor also it is like that, everywhere it is like that. But in case of solid state, you don't have to do like that. You can assemble a single can all this part, because when there is no liquid part, the house number 1, the separator number 1 doesn't know what the separator number 2 is doing or separator number 3 is doing, because they are isolated, they are not interconnected, they are separate from each other. But when it is liquid, and if you put like this, everything will get interconnected. So that is the benefit of solid state. Okay, so we now come on to the capacitor technology. I will have to go fast forward. See, the capacitor technology and the supercapacitor it is actually a capacitor. I would rather put up in a way a normal electrostatic capacitor, which you ever know is a first generation capacitor. The electrolytic capacitor.
Sir, I'm sorry to interrupt, but you have 5 minutes more.
Okay,
So,
okay, but actually I'm almost yeah, because see, my sometime was eaten up earlier.
Yes,
Okay,
yes.
and
Yeah, right.
and I'm really I'm not even I'm just trying half the way now. I will try to speed up. So I request you to slightly, yeah, because okay, so
Okay, sir, continue.
yeah, please. Because after that, I think we'll get charged up by end of this session, and after that there's a lunch time, so we can just everybody can accommodate that part. So
yes, we can store some energy then.
Yeah, sure. So I'll just try to skip off many things now. Within 15-20 minutes, I will try to finish it up by 12:30. So whatever time was allotted, I will be completing in that, because I started late. Anyway, this is the screen. You can see the construction of supercapacitor. Normally, it is looking identical like lithium-ion battery. So far, the material is concerned: anode, cathode, separator, and electrolyte. Lithium-ion battery is also like that: anode, cathode, but the material, of course, is different. So we'll just go as usual history. The history of supercapacitor dates back to actually 1957. But you know, the best part of it, until 1970, nobody knew it is a supercapacitor. So that is the great part of that. And after that also, after 1970, not much development had happened in supercapacitor. But in last decade, in last 10 years, lot of development has happened. We'll cover up that. So to understand the things very specifically, on your screen, you can see a convectional capacitor, 2 electrode and a dielectric, that is polymeric capacitor or ceramic. The other one is a metal oxide or electrolytic capacitor. If you notice the yellow part, which is dielectric, which is going smaller and smaller. Okay, first it was big, now it has become smaller. Now you can see the supercapacitor on your screen. It's a thin line. Because in capacitor, you have only one option. To be honest enough, not one, two options. When you want to increase the capacity, storage capacity of capacitor, your farad value should go up. Okay, what is farad value? C. C is directly proportional to area of the conducting plate and inversely proportional to distance between the two plates. Now, if you increase the area of the plate, your capacity will go up. If you decrease the distance between the two plates, again your capacity will go up. So whatever advancement in supercapacitor is there, it is based only on 2 points. One, to increase the surface area. Second, to decrease the distance between the two plates. And this portion I will give some slight time to make you understand in very simple way what is supercapacitor. That is very important. Otherwise, there is no use of the lecture. Okay, now suppose I want to increase the area of the plate. The nanotechnology has come into has a blessing actually. Everybody know activated carbon. Activated carbon, particularly made for supercapacitor. The surface area of that activated carbon for 1 gram. Listen, for 1 gram is almost 1,000 square meter. 1 gram of activated carbon will give you a surface area of 1,000 square meter. So what we have done, we have coated this activated carbon on aluminum plate. Great. We have got a big surface area in a small plate. The second part, distance between the two plate. We are not using a physical dielectric. We are not using a physical separating part. Whatever separator is used in supercapacitor is for different reason that you will understand from the diagram. And what is happening, we are using an electrolyte, and that electrolyte at particular when you apply the voltage, it forms an insulating layer at the surface contact area. This particular phenomena is basically known as Helmholtz layer. I will try to enlarge this picture. Yeah, it is known as Helmholtz layer. What is the Helmholtz layer? It is forming an insulating thin imaginary line, and we are using this insulating part as a dielectric. Okay, and you can see there are two capacitors formed in series in this single capacitor. There is an equivalent diagram. So now rest of the part you can Google it or refer a website. You can, sorry, oh my God, what has happened? Yeah. Okay, now how many types of supercapacitors are there? There are 3 types: ADLC, pseudo supercapacitor, hybrid supercapacitor. I will just not explain detail into that. Okay. Asymmetric also, you can Google it, you will find what is asymmetric and what are the electrode part. This also will cover it, go fast forward mode. And we have also made graphene. Like I told you, activated carbon gives you 1 gram, 1,000 square meter. Graphene gives you 1 gram 4 times. That means 4,000 square meter. But other property of graphene is there. You don't have to use 1 gram. You can use a 2D material. You can use a thin layer. So again, that will increase the energy density. So potential application, these are whatever you think supercapacitors can be used over there. I will go fast forward on to that. Now this is a very important part. Now please don't misunderstand. The batteries and supercapacitors are trying to compete with each other, because competition is always there when you have identical properties. Okay, identical properties. These particular two technologies are having a complementary resemblance. Whatever is available in supercapacitor is not available in battery. Whatever is available in battery is not available in supercapacitor. Now this both the technologies are in evolution mode, and lot of things are happening, and it is happening, trust me, on only one front. Batteries, the technological development is only on one area. They want to behave more like batteries. Sorry, the batteries want to behave more like supercapacitor, and the technological development in supercapacitor is like they want to behave more like batteries. So they are converging technologies. They are trying to converge into each other, and the day will come when you will have something. You may not. You may call it as hybrid supercapacitor or hybrid battery. The time will tell. Okay. And there are challenges. I will skip up this. All the challenges are for two batteries. The requirement is any power device or any electrical part, they require two type of energies, power density and energy density. In a nutshell, batteries are good at energy density, supercapacitors are good at power density. So you require both. And what you do, you have one technology is not able to support you. This experiment part I will just forward with to prove. Okay, uh, any individual technology is not able to support you on that, both the part, power density and energy density. So you call it as it is known, necessity is mother of innovation. You require both, but where is the mother? Okay, we are into frustration. Okay, frustration is father of creativity, and in Hindi, the creativity is known as jugaad. So whatever is available presently, you combine best and do something jugaad, that is known as hybrid in English. Okay, so we have done some trials on hybrid part, like combining supercapacitor and battery. This is done on lead acid part, demonstrated practically hybrid energy storage, validated by ISRO, Indian Oil, and some scientists. Okay, and we have also made a four-wheeler with the hybrid technology. In fact, you can just see the video of this hybrid vehicle. It is not petrol or diesel. It is a pure electric vehicle, Tata Safari, made by us. And to demonstrate the capability of hybrid energy storage, we have used the batteries and supercapacitor into that. This is a big vehicle weighing 2 tons. Okay. And see, the engine has been removed. There is no engine. All batteries are there. These are lead acid battery, and supercapacitor is there also. Okay. I'll just, yeah. These are the supercapacitors. Okay, we'll go forward into that. You can use the hybrid system in solar also. If you use battery and supercapacitor combined, the battery size goes down by 40%. The run time, your backup time goes up by 50%. In the solar, in windmill, it is 57%, and the battery life goes up 2 to 2.5 times. How it goes up? Because I will just take a short time to explain that. Because with the battery comes with a life tag. Like a lead acid battery, you have 300 charge discharge cycle. After 300, the battery is over. In lithium ion, it starts from 500, goes up to 800, 1000, depending on the chemistry. The best part is available in lithium titanium oxide, which is around 3000 plus. Okay. So by if you use supercapacitor in parallel, what you are doing, you are postponing the work of life cycle of the battery, because the life cycle is counted then in the terms of deep discharges. When the time comes for deep discharges, your one cycle is over. So if supercapacitor is taking care of the deep discharges, discharging, the battery is saved from that part. So your life of battery goes up. So this is a combination of lithium ion battery and supercapacitor with electronic support. Okay. Now I'll just go slightly the future part also. Like we are talking of hybrid from external. Okay, we have done hybrid from internal also. We have made a lithium ion supercapacitor. What is that? The figure is visible on your screen. One electrode of supercapacitor. We have taken one electrode of supercapacitor. We have literally robbed one electrode from lithium ion battery. We have assembled it together, jugaad kiya hai, or English more respectable word, hybrid, and we have formed a lithium ion supercapacitor. Now with this, we have combined best of both the world, and it is a single component. The energy density has gone up. The charging time is of supercapacitor. The energy density to some part is of battery. Okay, so we have scored ahead. So the things will go on improving and improving. And basically, as my promise, okay, I've tried to finish it on fast forward mode. Okay, so may you all stay fully charged. And thank you with this. Thank you, ma'am. And I'm done.
Okay, sir. But the talk was very interesting, and it is a very wonderful experience being with you, because you are the real entrepreneur, and you have been inspiring our young entrepreneurs and the researchers who will, in their future, who might want to go in your path. And rightly, you have said that Atma Chintan to Atma Nirvar. So that is a very rightly said thing, and you have explained the evolution of the energy storage systems from Daniel cell to lithium-ion batteries, both in liquid and solid state, and then to supercapacitors. So many people, I think, they are working on such kind of devices when there is an energy crisis now, which is existing globally. Thank you, sir, for such a wonderful talk. And some people, I see some people putting some questions in the chat box. Probably I'll try to take one or two of them. So it is a question from Sunny Nandi. He wants to ask that though the solid state batteries show promising results, but their resistance at the electrode or solid electrolyte interface is too high, hindering fast charging and discharging. So what is your opinion?
Yeah. See, as I told, that is a matter of research. In fact, we have made that solid electrolyte because he is referring to polymer part. In polymer solid electrolyte, that resistance is high, and the battery is losing on power density. But ceramic and glass, we are working on glass also. The results are very good, and that's why it will take another 4, 5, 7 years for that to come out, but there are promising results into that, and fast charging will be possible. There won't be any heating effect also, so you be rest assured. Whatever you have referred is actually related to polymer, but lot of improvement is going on in that. It is the initial stage. Yeah.
Okay, thank you, sir. Another one question I'll take, that is from Nilaya Mahanti. Wants to ask that can we have supercapacitors by simply inserting a dielectric material with a high dielectric constant in between the two plates, rather than changing the surface area or distance between the plates?
No, you can't do that. You can't do that in a sense. See, for everybody, I would rather put up in a way, what is 1 farad? 1 farad is 1 volt, 1 ampere for 1 second. Listen, 1 volt, 1 ampere for 1 second. So this is a storage. And if you want high storage, you want high farad capacity. And if you are inserting physical dielectric, the dielectric what we are using, the distance between the two plate, in a supercapacitor, I will tell you, it is actually 2 to 3 Armstrong. Then only we are able to get that kind of use. So there is no physical dielectric because available. You cannot see that kind of thin dielectric, and you cannot wound that dielectric. You cannot physically handle that dielectric. So if you use physical dielectric, it won't be a supercapacitor. It will be a normal capacitor. Yeah, because everybody must have seen a fan capacitor. The fan capacitor is 2.5 microfarad. So what we have done, in the same size, in the same weight, we have multiplied the capacity by 5 crore. Imagine 5 crore. So that cannot be achieved with physical dielectric. Okay, I'm done with that.
Okay. Thank you, sir. We are glad to have you with us.
Thank you. Thank you so much.
There are no more questions. So may I now invite our next speaker, that is Dr. Amya Priyam. So I think by now everyone is acquainted with him. He has already delivered 2 talks. still a very brief introduction. He works as associate professor and head in the Department of Chemistry in School of Physical and Chemical Sciences in Central University of South Bihar, Gaya, since September 16, 2013. His broad areas of research are luminescent nanomaterials, quantum dots, subconversion nanocrystals, plasmonic nanomaterials, that is hollow gold and silver nanoshells, silver nanopyramids, photocatalysis, plasmon and exciton based, SIRS based sensing inorganic oxoanion and biomolecules, it is dark field and luminescence imaging, photodynamic therapy, and photothermal therapy. Now he has a research experience of many years. He worked as a postdoctoral research associate at the Department of Chemistry, Florida State University, and also he worked as a postdoctoral research fellow in the Division of Bioengineering, National University of Singapore. He also worked as assistant professor in the Department of Applied Chemistry in Birla Institute of Technology, Mesra, Ranchi. He has already handled 3 projects from DST, Nanomission, Fast Track, and CRDS grant. His publications number 243 and his total number of citations is 859, grabbing an H index of 16 and I-10 index of 22. Now today he'll be talking on soft chemical routes for tunable plasmonic nanomaterials and some interesting applications thereof. Sir, please, I welcome you to this session. You can please start your talk.
Hello, am I audible?
Yes, sir, you're audible.
Okay. And I hope my slides are visible too.
Yeah, you can just bring it to slideshow mode, I think.
Yes, done. So, as the title suggests, I'm going to talk about plasmonic nanomaterials. And that plasmonic nanomaterial, let me tell you what they mean. What I mean by saying that there's certain metal nanoparticles which show a property which is known as surface plasmon resonance, and so all those nanomaterial that show this kind of surface plasmon resonance, due to which they have a variety of colors can be seen when they are reduced to nanoscale. So we call them as plasmonic nanomaterials. Usually they are noble gold, like noble metals like gold and silver. And so I'm going to talk about, and the tunable term here is coming because we are normally, if you synthesize spherical nanoparticles, there is not much. You see a typical yellow color for silver, a typical red color for gold, but we need to move out from this visible range to near IR range for several applications, as I will discuss as I go ahead. So the term soft chemistry here refers to techniques. The synthesis technique used here is we employ mild reactants, mild conditions. So there's no extreme heat or extreme cold thing is needed, or extreme of pressure is needed. All the conditions used are very mild. So coming to this, so normally if you visualize a metal such as gold, as shown over here, let me. So normally, if you remember metallic bonding, we know about it. So there is a metal lattice, cation lattice, that is submerged in the sea of electrons, and the sea of electrons is symmetrically distributed around this. If the shape of the particle is spherical, the sea of electron is symmetrically distributed around this metal kernels, that metal ions. Okay, and so that is the case when symmetrical distribution of electrons will be there when you have when there is no electric field applied. Once you apply electric field, as shown over here, the electrons will move. If the direction of electric field is this one, then the electrons move to this side. and in the opposite direction to the electric field as shown here. So you have excess of electrons on this side and deficient side is here. So a positive charge dominates here. So a dipole is created. Now in the next cycle, this is this sinusoidal variation that I am showing that is of an electromagnetic radiation, the light. So when in the next cycle, as the direction of electric field is changed, the dipole, you can see the electrons move to the other side. So in this way, rapid oscillation is set up. There is a strong absorption of light at a particular frequency. This is it is characteristic. The characteristic frequency varies from metal to metal. For example, spherical gold typically absorbs around 500 nanometers, whereas a typical silver nanoparticle will absorb around 400 nanometer. If we so this surface plasma on resonance can be put to use in various applications, like catalysis, like sensing, and also in for therapy. So I'm just but this spherical, if you talk about normally when we synthesize nanoparticle, it is spherical shaped nanoparticles of gold or silver is synthesized, and it is found that there is a limitation to the optical properties that they can have. They have what is shown over here is that when you whether you make a 10 nanometer size gold nanoparticles, and then slowly if you go on increasing the size of nanoparticles, they have you can go maximum change can be 580 nanometers. Okay, so by increasing the size by 10 times, only 60 nanometer change in the absorption is observed. And this is not useful for applications because I'll just come to that point. Similarly, there is also limited tunability in case of silver nanoparticles. If you simply, if keeping the size spherical, if you change the size like 5 to 100, you will have only a 390 to 460 nanometer change in absorption peak. So why I'm saying that there is a need to tune this SPR wavelength is that the SPR frequency that we have just seen, it needs to be tuned to a desired wavelength for different types of application. For example, the motivation here for our work is that for therapeutic purposes, it is required the prominent mode of therapy in these nanomaterials, the plasmonic nanomaterial, is what we call as photothermal therapy. And this is a very good approach in the sense that it is non-invasive in nature. You don't have to make incision or do a surgery. You simply have to inject and then shine a laser to the desired area where cancer is there, tumor is there located, and by shining a laser, you can light will be converted into heat, and the tumor will be gone. So this type of thing is good, but if you are using normal gold particles, they absorb in the visible range, and the visible range, our blood is red in color. There are many more proteins and amino acids which absorb in the visible range. So when if I am using a laser, of visible laser, it will excite all those molecules in the body, and there will be unwanted damage to the healthy cells. So this has to be avoided. When I say non-invasive, it should not affect all these things. So it has been found by analysis that if we tune, there is an optical window from 650 to 1200 nanometer, which is transparent to the from biological point of view, that is none of the cell organelles, none of the biological components absorb in this region. They do not have, they have negligible absorption. So it can be said to be transparent in this region. And this is the window. This is the optical window, spectral window that we need to utilize for this kind of light-induced therapies, light-induced diagnosis, if we want to make them non-invasive. So to make them non-invasive is the theme, and that goads us, that propels us to tune the SPR frequency of the gold or silver nanoparticles to this particular region. It has been further analyzed in recent papers that this range, NIR range, suitable range for biomedical application, is further split into 3 sub-windows, NIR sub-windows, that NIR 1, 2, and 3. So that it is a recent analysis of this spectrum, which we can see here. So the 650 to 950, then 1000 to 1350 nanometers, and then 1500 to 1750 nanometers. So this is the range that we should be looking at if we want to do a laser-induced photothermal therapy or things like that, diagnosis based on that kind of thing, which is possible. Like we have dark field imaging is also possible using these nanoparticles. So for all these purposes, to make the diagnosis as well as the therapy non-invasive in nature, we need to tune the this SPR frequency, SPR wavelength into this region. So this is the requirement. But as I told you, that if you only increase the size, we are not getting it there. We are not able to tune the SPR wavelength, the plasmon wavelength, to the desired suitable region. And so we look for the ways, the means, how we can tune the plasmon peak. So there are existing methods. If you carry out a literature survey, then there are suitable ways. There are 2 principal ways that I can see. There's one is that change the shape. Don't keep it spherical. Change the shape. Make pyramids, plates, or anything. Nano rods. You can have. So if you can make nano rods, it is possible to go into that region, that desired range that we talked about. 650 to 1200, that was so we are going into that. We are also going into nanoplates like this, you can see. So here also it is possible. So this was the prior state of the art when we started our work. And so here the tunability comes from the splitting of the plasmon modes. There are 2 plasmon modes. One is oscillation of collective oscillation of electrons along this axis, collective oscillation of electrons along this axis. So there are 2 plasmon modes. One is constant, as you can see here, and the other absorption, longitudinal one, is tunable. So next, the second approach is that keep the shape spherical, no problem. But suppose if you keep the face spherical, is it possible to get it into the desired range, to the NIR region, near IR region? Yes, it is possible by changing the structure. We keep the shape spherical, but make it hollow from inside. And as you can see, as you can change the size of cavity, the void space inside, we call it aspect ratio, the outer diameter to inner diameter ratio, shell thickness. And if you have outer diameter to shell thickness ratio, that is aspect ratio. So as you go on increasing that aspect ratio, you can move to the desired range. It is possible to redshift the plasmon peak. You can move to a longer wavelength side and reach into the desired near IR wavelength for the therapeutic activities desired, which will be non-invasive, and also the light to heat conversion efficiency will be higher in that region. So so this is the area that first I am going to discuss. I'll be discussing both the methods approaches which we have followed in our labs. But first one is the hollow nanoshells that we have made. So the prior state of the art at the start, when we started our work, was this. Silver nanoshells the methodology to make silver nanoshells was very primitive. It is not. There was no tunability in size to that desired range. It was only absorbing around 500 nanometers, and even if in some other works we found that the peak was too broad, again not suitable for biological purposes. When you want a very precise wavelength to be used and want to have a very high absorption at that particular wavelength. So this is again not suitable for the purpose, the application that we intend to do. So we designed our own methods, and this is what I remember in the first talk. Dr. Shrikant also talked about nanoscale Kirkendall effect while talking about hollow magnetic particles. So this is also a nanoscale Kirkendall effect, which we have modified, and it is a different way put it. We have in this we have we make a silver oxide nanoparticle first, using the typical reagents like silver nitrate, and NaOH, that is sodium hydroxide, and then use a mild reducing agent. This is hydrogen hydrate, which is a mild reducing agent. And so looking from the reference frame of Ag2O nanoparticle, what happens, there are 4 things that is going to happen. The Ag2O will dissolve and it will move outward. Ag2O will dissolve and form Ag plus ions, and Ag plus ion tend to move outwards. So this arrow shows that point that it is going outward. And when you add this hydrogen hydrate, it will move inwards. So this way, at some point where the two meet, the Ag plus ions will travel outward, the reducing agent is coming inwards. So there will be a meeting of the two at some point, and at that point you will have Ag naught, that is silver atoms will be created, and collection of silver atoms in a series in a lattice-like pattern will give you a hollow nanoshells. So this is what is we by controlling these two types of diffusion, inward, outward diffusion in a very precise manner to achieve desired thickness, is what is at the heart of nanoscale Kirkendall effect. So that is what we have worked on, and we developed. So these are the events that matter in this thing: outwardly mobile reaction front composed of dissolution of Ag2 and outward diffusion Ag plus. I just talked about that in the previous slide. And there's inward diffusion of this hydrogen hydrate, and where these two meet, there will be reduction. So these are the parameters which will affect the formation, a delicate interplay of these things: the outward mobile reaction front and the inward mobile front will create there's a delicate interplay between the two, and this when the two oppositely moving reaction front meet, there is a formation of shell-like, hollow shells-like structures. So what we found, that order of addition is very important in this case, and it matters a lot whether you are adding reducing agent to the silver oxide or your silver oxide is added to this reducing agent. So depending on that, we have classified, we evolved methodology, normal addition. When we call it normal addition, when we add the silver, the hydrogen to the Ag2O, silver oxide, this is called normal addition. And within this also, you can have two types of methods, like single shot addition, very fast, and there is fast addition, and this is slow. You add it drop wise. So we call it slow normal and fast normal. There are two types. Similarly, in the reverse addition, when we are adding silver oxide to the reducing agent, we call it slow reverse and fast reverse. So we found that fast reverse process, the fast single shot addition of the reagents in the reverse manner is better. It gives a better tunability. And we can see that in the HRTEM image shows us a hollow structure. So this was the first hint that we got that we are going into the right direction, and such nanocrystals. And we also developed an empirical scaling law. Empirical scaling law means that we are we can extrapolate at that what is the desired aspect ratio for a particular for the shift of SPR peak to a particular level. So that's what this is all based on experimental values. So we can see that an aspect ratio 5 gives us around this 8 beyond 6 around 600 nanometers of the plasmon wavelength. So this is another variant of that nanoscale Kirkendall effect, and here we have used folic acid in the previous case, we have used the citric acid as the capping agent. Here, using the folic acid, I can also tell you that the 4 parameters that I told, they are affected by temperature very much, all of them. So it is very crucial to optimize the temperature. So in the previous case, when we are using citrate, the temperature was around 20 degrees C, where we get optimum peak positions, like shifted to the red shifted and narrow peaks. In this case, it was because folic acid are slightly larger molecules, they move slowly. We had to heat it to up to 50 degrees, slightly higher than room temperature. And then we found that we are getting the hollow structures. And we could also prove, so these are the TEM images, and we also suggested a mechanism which was in this, that is, we call it void collisions mechanism. So all the details are in the paper, and we got this proof of our suggested hypothesis in form of TEM images. You can see that there's initially small voids which are formed in a crystal, and these voids fuse together to give a larger void in the center. So this is the mechanism of the Kirk-Dial effect that is prevalent in the synthesis of hollow nanocubes of silver. These are the X-ray diffraction, which shows phase purity, and this is the tunability that we have got, which I was talking about ultimately. So by changing the temperature and stirring speed, we are able to tune it to up to 630 nanometers by varying the condition as shown over here. By temperature, so 50 degrees Celsius was found to be the best. It was also, it was a big chain of experiments actually, which led to identification of a suitable ratio of reducing agent to silver ions, because it is not easy to get the right kind of hollow structures in one shot. It is you have to optimize the things. So there was optimization of stirring speed, and we found that there is mild stirring. You don't have to stir very hard, very fast. You have to go do only a mild stirring to get a nanocrystal, hollow nanocrystal with red shifted peak, as well as a narrow peak. Narrow means in a spectral sense, we talk about FWHM, full width at half maxima. So that should be less. That is the parameter we fix for high quality nanocrystals. So these are the optimization that was done for this folic acid capped hollow silver nanocrystals. And here again we have made a scaling law, empirical scaling law based on our observations. So what will be the diameter ratio of inner diameter to outer edge length in this edges? So that we worked out, edge length versus SPR shift. This has been worked out. We also calculated the extinction coefficient. This was a challenging task to calculate the extinction coefficient of each of these nanocrystals. And besides that, but what we noted here, that even after doing so much effort, we were not able to move into the near IR range. And so we further strategized, although the tunability that we achieved was highest, if you compare all the other methods available, nobody in the silver nanocrystal, hollow silver nanostructures have shown the tunability as of that. But we were not going into the near IR range, and still the challenges was not yet overcome. So there was another modification of the work that was done, and this is something that we were successful, and we have patented it. We filed a patent for this. This was again near IR. We were successful in making near IR plasmonic nanoshells of silver, and the nanoshells were synthesized by tweaking the method a bit from the earlier one, and we had used dimethyl glyoxime as the capping agent, and slight variation of the same pattern, and some temperature used here was around 5 degrees Celsius. So under those conditions, again there is Kirkendall effect that occurs. We have a void collisions, and we have formation of then the void, because of the collisions of void grows, keep on growing, and you have a larger void as compared to the initial template that was used. So this is a void collisions mechanism again over here, acting over it. And so this is so now if you look at this, the phase purity is there. So there's also one more important thing that we patented is the method to produce. Usually when you go to the synthesis lab where nanomaterial, you have to synthesize nanomaterial, it is always advised that you keep studying your solutions. You must mix it homogeneously and keep it studying very well. So only then you will have a narrowed size distribution. A nice peak will be observed. A nanomaterial will be formed. But we are showing that there are certain synthesis methods as in this one where stirring is bad. You don't have to stir the solution at all. We carefully analyzed this system and we found, as you can see here, there are the different stirring speed, 0, 180, 5, 360, 1350. We found that as we reduce the stirring speed and finally go to the 0, we get a very nice peak into the near IR region, which was desired for biomedical application. 920. So this is the kind of nanocrystal that we need, which have a less FWHM and a red-shifted peak to the towards the NIR. So we are clearly into the first NIR window now. So this was we further challenged ourselves. Can we go further and tune it further? So even this peak that we are seeing here is the first to be reported in case of a hollow silver. Hollow silver, this is the highest that we have achieved in the so nobody in the world has so far done that, and we even challenge it further ourselves. And so when I say high quality nanoshells, what are so the identifying features are phase purity. Well, XRD we have done. Intensity and tunable plasmon peak, that's you can see the maximum tunability we have got. We have also got enhanced intensity. As I showed you in some previous examples of the published papers earlier, when they are going into the near IR region, the peaks were not good. They were typically broad. So we are getting very intense, and FWHM is less. Tunable good crystallinity is there. So that was verified by HRTM imaging. So narrow size distribution and uniformity of shapes. All this thing was there. So now if we classify now, this is by adjusting the few parameters more critically, we were able to further tune it to the 1000, beyond 1000 nanometers. You can see the maximum tunability we could achieve was 1150 nanometers, and you can see we are not able to take the spectrum on this side because at around 1400 the water starts absorbing. So that is a problem. We are trying to fix that, and we will be further trying to do it. In the NIR3 window is still unexplored, and that is something that we will be doing it. So these are the, as they say, seeing is believing. So here are the TM images. So you can see here the sizes, outer diameter 58, 53, 50 nanometers, around 50 nanometers. There are different aspect ratio, shell thickness is different. So, and the maximum shift that we have got is in the 40 nanometer shell, which has an aspect ratio of 5. So higher aspect ratio, your greater is the red shift. Higher is the aspect ratio, greater is the red shift. That is valid. We have observed through this experiment. We can conclude from these experiments. And so that was the high resolution images of single nanocrystals, hollow nanocrystals, silver nanoshells. And this is the image that shows you the distribution profile. You can see all of them are showing the hollow pictures in all the 5 images. And the distribution histogram, size distribution histogram was generated. This is and by FTIR, doing FTIR, we can confirm that DMG is capping on the surface. It is lying on the surface. So So we analyzed the FTIR. I'm not going to details of it.
Sorry to interrupt, but I think we have to finish up earlier. Fast.
Okay, give me 5 minutes. I will finish.
Yes, sir.
Okay.
You can take 5 minutes and wind up.
Okay. So similarly, so these were the hollow nanoshells, and I talked about by changing the structure how we have been able to tune it to the near IR region. We also were able to tune this plasmon peaks by changing the shape, and this is our method that was developed, which was seedless method. Most of the previous method used seed nanoparticle and then grow an anisotropic structure. Here it is a single step process, direct synthesis, which involved the tuning of hydrogen bonding interaction between reducing agent and the capping agent. So by this way, we were able to form spherical nanoparticles, nanoplates, nanopyramids. So gradually you can have, so these, you can see the beautiful colors as we go into the near IR range up to the 1000 nanometers. So clearly, so by all both the approaches have been utilized in our lab to synthesize silver nanocrystals. Particularly hollow ones were challenging, and so these are the pictures that you can see. Pyramid-like structures are there. You can see the atoms arranged in a crystalline pattern, lattice-like arrangement. So these are the things we analyze performing second derivative analysis. We could analyze the hydrogen bonding interactions also between the stabilizer and reducing agent, and then we form empirical relation was also found out. So you can see on the y-axis we have plasmon peak shift in plasmon peak position versus FTIR intensity ratio of the hydrogen bonded CO and non-bonded CO. So that gives a measure of extent of hydrogen bonding interaction in the system, and we could establish that as the hydrogen bonding increases, the plasmon peak is shifted to the red side, and we are able to move to this. So now I'm moving into the applications. By using this anisotropic nanoparticles, pyramid-shaped nanoparticles, we were able to develop a method for detection of arsenic in water. And so the sensitivity, this is the method which we call as surface enhanced Raman scattering. It is so you can see here, in case of simple arsenic in the water, we are not seeing any peak, but when we add nanoparticle, we are getting intense peak over here. And when we plot it with different concentration, this is our calibration curve, which shows this thing. And similarly, we made chromium 6, chromium 3 present in water that can also be analyzed. And the lowest detection limit is 30 ppb, 40 ppb, and so on. So these are the methods. And the second application is catalysis that we did using these hollow silver nanocubes. We can show that it can act as a catalyst in presence of sunlight. That is, you can see, and also weakly it is a weaker catalyst in the dark, but it is a catalyst. So it is a different method. The model system here was taken of that was of methyl orange, and the degradation was catalyzed by this silver nanoparticles. So this is a method which, and we could also establish that as we move into the near IR range towards the red side, sorry, not at 630 nanometer, the nanoparticle with 630 absorbance, the 630 nanometer SPRP acts as a better catalyst as compared to nanoparticle which absorbs at 400 nanometers. So clearly moving into the red makes sense even in catalysis. It is also important to note that all these catalysis that we are doing was done in sub-picomolar regime. Generally, it is a microgram or a nanogram amount that is added of catalyst that is in powdered form are added. But here we are showing that even the catalysis, our catalytic system is so good that catalyst is so good that we can use only sub-picomolar amount to get the desired product. The recyclability was also good, and 3 cycles also there was only a little reduction in the rate constant, as we can see. And this is the latest work that we have also been able to establish with help of pharmacy group in the BIT Misra, where we use these nanoparticles, the pyramid-shaped nanoparticles, for which were capping agent were changed to folic acid, and it was studied in the tail when injection was given to the mouse, and it was effective in getting rid of the cancer cells, which was shown in the result. It has a dual, so it was a dual effect of chemotherapy, targeted delivery, and photothermal therapy. So by doing so, a very good system was developed, which which can specifically target the cancer cells because of folate moiety on the surface, and because of the quercetin. This is a flavonoid which was attached to the surface that was delivered to the tumor site, which also acts supplements that therapeutic activity, and so the side effects are minimized, and we have a good system. And so we are also in the process to analyze our hollow structures, which are yet to be done in this context with respect to cancer treatment. So that would be our future work. And so I conclude here. So there is so the methodology, it is possible used by us, develop that is using mild reducing agent and mild reductant. This approach is very helpful in getting any type of structures. By using the same set of reagents, I am able to produce hollow nanostructure. I am also able to produce anisotropic silver nanostructures with tunability of plasmon going into the near IR1, NIR2 region, as I showed above in the previous slides. And so it has been developed based on those nanomaterials. We have been able to do surface enhanced Raman spectroscopy based detection of arsenic, chromium to a very low level of 30 ppb. We have also been able to do the catalysis. We have been able to show that if going into the red is good, towards the red side, NIR side, the plasmonic catalysis improves further. So this plasmonic catalyst is a new class of catalyst that is coming up in this field, and this is very exciting, because if you can utilize sunlight. Remember, sunlight has around 40% of sunlight is IR, near IR. So it is important to utilize that part of the spectrum as well. In that case, so all this leads to a greener, cleaner, effective methods for for producing the nanoparticles, and non-invasive technique. Based on these nanoparticles, we are also able to develop non-invasive techniques for cancer treatment. And with respect to hollow nanostructures, we are yet to do some more work on cancer treatment aspect. Even diagnosis aspect is important. So those are the things that will be our future research, future work that we will take up. And these are my group members. Bhavesh, so he's familiar in KIIT. Bhavya Bhushan, Shatrupa Patnaik from VIT Misra, she was my first PhD student, and Dr. Shah from UGCDA CSR, he was my PhD guide earlier. And the funding must be acknowledged, because that is keeps us going in the research area. So Nanomission, SERB, and UGCDA CSR, they are gratefully acknowledged. We thank them for providing us the funds. Thank you.
Thank you, sir,
thank you for
such an informative talk. Sir, you have given very nice information regarding your research work, where you have used some novel synthesis technique using nanoscale KIIT Kendall effect to prepare hollow silver nanoparticles, and also some pyramids shaped nanoparticles, and the application in photocatalysis, as far as I know, visible light photocatalysis is a major area of research now, and people are trying to do research on that to replace the UV.
So we are taking it further, moving to the near IR, taking it further to the near IR.
Yeah, that is very much necessary also for creating a greener environment. Thank you, sir. And due to lack of time, we are just not taking any questions now. In case of any questions, as I see there are 2-3 questions in the chat box, you can just drop down a mail to the speaker so that he can answer your questions. Now we end the session. The next session will start at 2 p.m. sharp, and we'll be right here together again for some interesting talks today, and also we'll have the valedictory session at the end. Thank you, sir. Thank you to the speakers today. So now we will have a break. So bye for now.
Thank you so much.